Good morning.
Good morning.
Good morning. Hello.
Hello, everyone. Bright and fresh new day. Sun is shining here. So very welcome, everyone, from around the world. Let us know where you're watching from, by the way, in the chats on the different places. And it was really cool and exciting day yesterday. I really loved the talks. Very, very impressive what people are building with their technologies and their data sets and graphs in general. And can't wait for today. How about all you two?
Absolutely, yeah, same. I had an exciting day yesterday. I think it was really good seeing all these presentations from all. We touched all various topics yesterday, and I think today it continues. I'm really looking forward to the session. So yeah, I think it's gonna be good.
I agree, completely agree. It was fantastic yesterday. We have exciting stuff in store for today. Let me share my screen and let's have a quick look at the agenda for today. So
let's do that.
There we go. Sorry, I clicked the wrong button. I wanted to click here. Perfect. So this is our agenda for today. So after a short opening from us, we have a last-minute edition, an absolutely interesting and fantastic talk about the clinical knowledge graph. I suppose a lot of you know the publication. It's a bioarchive, and Rita will describe this graph-centric framework for translational clinical proteomics. And from what I know about the clinical knowledge graph, the scope of data they've integrated is massive. And the interesting thing is the whole framework to build this knowledge graph is public and available. We will follow with a super interesting talk from the German Center for Diabetes Research, an interesting and also very, very public and very open knowledge graph project with a huge focus on publications and integrating everything in a translational manner from clinical research down to basic research. We'll switch focus a bit to microorganisms, which is, I think, super interesting. We have a talk about Neo4j for bacterial genomes, and I'm really looking forward to data models and how Xing models a genome and what kind of queries it can run. We will follow with a talk on multi-omics integration, and I think this topic is very relevant and is interesting for almost everyone. And what I found particularly interesting yesterday is in the OMOP integration talk, there was also multi-omics integration kind of in the future. And one of the motivations why they want to go from the OMOP system, OMOP Common Data Model, to Neo4j was to be able to integrate non-patient data, or let's say non-observational data, like multi-omic studies of those patients. So while this is also about microorganisms, multi-omics are multi-omics, like independent of the actual organism, right? So this is going to be an exciting talk. We will follow with a bit of research. David Motin from Aarhus University has maybe one of the best titles here, Unveiling the Knowledge and Knowledge Graphs. And what David does is he has a huge amount of graph algorithms and graph data analysis methods available that he can apply to rework knowledge graphs. And while the GDS is impressive and growing, there's so much more beyond that we can use to get more insights from our knowledge graphs. We'll get back to more healthcare orientation. Seshan Gori from IQVIA is going to describe this knowledge graph that they use for evaluating drug safety data with graph databases. And I think this is a very interesting topic because this whole idea of understanding what a drug does after clinical studies, after clinical trials is really growing. So everything in the area of real-world evidence is just very, very relevant for the industry, but obviously also for patients in clinical practice. We'll finish with a talk on natural language processing. And in the lifelike presentation from Evelyn Travnik yesterday, we've seen how powerful NLP can be in combination with a graph, because all the output and methods of NLP essentially produce graphs. And if you can get it into a knowledge graph, you can integrate it with so many different layers of information. And Emre will present his Spark NLP pipeline and how he creates a clinical knowledge graph on top of that. We'll have a closing session with also a bit of outlook, because like we mentioned maybe 500 times yesterday, the goal for this workshop is to foster collaboration and network. So So we want to bring researchers and industry together. We want to bring the healthcare space, the life science or research space together, and really fostering close collaborations, network, sharing of data models and data is, I think, one of the main goals of this event. Okay, that's our agenda for today. And without further ado, let's switch to Rita, who's going to present the clinical knowledge graph.
Hi, good morning, everyone. Can you hear me fine?
Yes.
Perfect. Yes, so thank you to the organizers for inviting me for this talk. I'm really excited to be amongst so many people that love graph databases and have worked so hard on them. So today, I'll just briefly talk about this framework we developed for translational clinical proteomics. It's based on a graph database, Neo4j database. Let's just see if it's working perfect. So we'll start with the why, the how, the package itself, the database, and the interfaces we created for it, and then where we envision the next steps going. So as you know, or many of you probably know, omics technologies like proteomics produce very high resolution data, which allows for a more holistic view of biological complex processes and complex diseases. And it's that exact same high sensitivity and high accuracy that allows us to search for biomarkers or protein signatures in health and disease, and use them hopefully in the clinic. The goal at Matthias' lab, both in Copenhagen and in Munich, is to make this technology cost efficient and high throughput, yielding quick, clear, interpretable, actionable, and reproducible results that can be more easily translatable into the clinic, and so closing this gap between research and clinic. However, this very complex and the sheer amount of data that is produced daily on multiple labs really highlighted the need for specific analytics tools to gather, mine, and integrate this kind of information, which we really found that was not so easy to get. So right now, a standard proteomics workflow can take hours until you actually get the spectra and it's finally processed. And after this, you would find other roadblocks like long statistical analysis that are not always standardized, so they can vary from lab to lab or even within the same lab, depending who runs it. Very time-consuming and unfruitful sometimes database searches and literature searches. And the biggest of all roadblocks is the actual manpower and time, as not all labs have access to bioinformaticians or other experts in downstream analysis. So as part of the computational clinical proteomics team, Alberto, myself, and Ana Laura, we set out to create a tool that would help standardize and automatize this downstream data analysis. So we basically took a page from Google and decided that a good first step would be to use a knowledge graph to help users resolve their analysis without having to navigate through other websites, having dozens of tabs open in their browser, lose days or weeks of constantly searching PubMed and Uniprot, for example. So we just wanted to make their life easier and save them time that they could be spending doing other experiments, for example. The CKG, our clinical knowledge graph automated framework, goes from MaxQuant or SpectroNaut outputs or other outputs like FragPipe or just MZ-tab files in minutes to actual knowledge. And this includes the automated analysis pipeline, so both the analysis and data visualization. We have libraries for all of this. It includes data integration as well, and not only with the metadata we collect from all these publicly available databases, but also if you have, for example, clinical data, proteomics, you want to get information or knowledge from both of them combined. So that would be in the data integration part. And knowledge mining, so sort of summarizing all the findings from the other tabs or the other analysis and showing them in a very user-friendly way, like networks or Sankey plots, and gather it with as well the metadata. So the workflow goes like this. We download these publicly available databases and ontologies. We have our experiments. If you wanted, also download some from Pride. And then we have parsers that basically transform all this raw data into very easy files to load into the database. So basically just nodes and relationship files with all the attributes we find relevant for the questions we will later be asking the graph. So this is all loaded into the Neo4j database with queries that we have predefined. So as long as these inputs have the same format, it can be loaded from other databases or ontologies. And then some other predefined queries that we created will also mine the database and feed that information into our analytics core module that runs in Python, but also applies R functions. All the plotting and analysis is dealt with in the background by a specific module, so we don't have to worry about it. Everything is predefined in the automated analysis, but you can access it in Jupyter Notebooks and manipulate the data as you please, and use other functions that are not in the automated analysis, which is also shown in a Dash app that you can very easily navigate. Yeah, so this is how it works. The database itself, as I said, contains a lot of other databases, 26 so far. We can always add more if we find that it's relevant to answer our clinical questions. But it includes UniProt, of course, PubMed, Drug Bank, the Human Metabolome database, Reactome, a lot of other databases, Phosphosite Plus if you want to look at modified proteins, and so on. Also ontologies, so we can standardize and harmonize all the knowledge. And that includes, for example, SNOMED, CT, because of the clinical variables and clinical data we want to analyze. Disease ontology and, of course, gene ontology for enrichments, amongst others. So these are numbers at the time we submit the paper. So now they are a bit higher, but at the time we had a bit more than 16 million nodes and 110 million relationships. I think now we are around the 200 million. And the data scheme is a bit more complex to, of course, the questions we would like to answer and solve, but also because we want to store all the information around the projects and the experiments from the researchers. So we actually attribute identifiers, unique identifiers, to both projects, subjects, and the samples associated to it. For now, it's analyzing mostly proteomics and clinical data. You can see the links to peptide, modified protein, and protein, but it would be very easy and straightforward to adapt it to other omics data, like transcriptomics or metabolomics, by creating the parsers and the links to transcript, for example, gene or metabolite. In terms of the interfaces, we have a few of them. They all run in Dash. We are looking into other more advanced ones. But for now, we have the homepage where we have basic stats on the Neo4j database itself, how many nodes, relationships, how many of them came from which database or ontology, the data scheme, also links for the other apps, and for the project reports themselves. So we also have a small clever search drop-down menu. If you basically type or start typing the name of your project, it will show you the ones that are available. And by clicking, you can just go to the report of the respective project. To make life easier for your users, we also created this project creation app. It's basically a form, you just fill it in, and then by clicking on the create project, it will create all these TSV or CSV files and load information into the graph database on its own on the background. And in the end, it will just give you the option to download these templates for the data upload, and also give you an internal project identifier that you also use for the data upload. In this case, for the data upload, it's another app, of course. You use the identifier. It will just do like a sanity check to make sure that there is the project you want to upload data to. You would select the kind of data you want to upload, the processing tool if we're talking about proteomics data, and then a drag and drop field for the upload of the files themselves. In the end, you upload the data to the CKG, and then you can run the default automated analysis pipeline just by clicking the generate report link. It will run everything in the background. It will retrieve the proteomics data from the graph database. It will run all the processing statistics, correlations in the background, and then in the end, you can go to the report app, to the report page, and it will give you the tabs for the different kinds of knowledge. There's a project information tab, a knowledge tab with the summarized information and metadata, multi-omics if you have more than one type of data, and then one for each type of the data itself. In the proteomics tab, for example, you would get ready for publication plots. They can go from QC simple plots like proteins per group, or peptides per group, coefficients of variation, abundance plots, to stratification plots, PCAs, t-SNE, UMAPs, functional stratification as well. The statistical tests, this will predefine or choose the appropriate test according to your experimental design. If it's, for example, paired or unpaired, if it's a t-test or an ANOVA, it will choose itself the appropriate one. And then, of course, the multi-omics, so correlation between, in our case, clinical and proteomics data in the form of networks or WGCNA, for example. So to illustrate how the CKG can accelerate both the analysis of the data and its interpretation, and potentially or hopefully support clinical decision-making, we use this user case from a urachal carcinoma patient with chemorefractory lung metastasis. It's a previous study we ran, and in this study, in the end, we proposed this KDM1A protein as a possible druggable target, which was approved by the tumor board. But we basically just ran the entire analysis, the default one. And in that case, we got more than 300 significantly regulated proteins, which is not ideal if you want to prioritize drug targets and candidates. So we extended this analysis in Jupyter Notebooks using only our analytics core and the knowledge we have gathered in the graph database. And so we started reducing the list of significant or relevant proteins by taking up only the upregulated ones. And from these, we focused on the ones that were already associated or were known to be associated to lung cancer by querying the graph database. Also, the ones that we reduced this further by selecting the ones that were targeted by inhibitory drugs, which gave us 19 proteins targeted by 60 potential drugs. And amongst them, this is just a subset of the network. Amongst them, we had KDM1A as a potential target, and this trenylcypromine as an inhibitory drug, which was the one approved originally by the tumor board for this patient. But we also had another drug that could also work as inhibitory drug. So already here we can see that in a very short, concise way, we could reach not only what was known and approved by the doctors and the tumor board, but other alternatives as well, or potential alternatives as well. So to reduce this list further, we actually wanted to avoid previously observed adverse reactions. So we retrieved all the side effects associated with these inhibitory drugs when used in these chemotherapy regimes. And we ranked them with a Jacquard index, and we filtered for a very low cutoff. So the fewer adverse reactions together with or towards these therapeutic regimes, the better. And from those 60, we selected 6. And we then looked in case they were also known to be used already in lung cancer treatments, and if from these they have by chance been described as combination treatments in the literature already. And to our surprise, then delight, we actually found trichostatin A and vorinostat that were mentioned in combination in more than 30 publications already. So in the end, we actually propose these two candidates as a potential treatment for this patient specifically. So this pipeline is completely reusable and can be applied to other studies. The functions are completely general, so as long as the inputs again have the same format, it could run a similar prioritization analysis for other studies. And this is only one of the many applications of the CKG. All the knowledge was mined from the graph database in a matter of a couple of hours instead of going and searching each one of the 19 proteins individually and looking for drugs that could target them. If I haven't mentioned before, by design, the Clinical Knowledge Graph is an open source community project, and we envisioned that the community could directly contribute to its further development, helping extending the database, the analysis, the visualizations, and notebooks. So if you are curious, please do go explore it, use it, contribute. Every aspect of it is very much welcome. The reports and the notebooks can be shared across multiple labs. So we hope that in this way, it will contribute to more open science. And thinking of long term, we also envision that having one knowledge graph connecting a vast amount of proteomics experiments could benefit the community immensely by allowing direct and deep project comparisons and leading to increasingly more robust and powerful analysis, and of course, all the knowledge generation that we could get from it. All of these opportunities would really allow us to support expert panels and medical doctors making clinical decisions, never replace them, but giving them a more thorough knowledge base in a faster and more robust way. And with this, we would really hope to move to an era of translational evidence-based medicine. For online resources, if you are interested, please go to GitHub. The repository is public. You can just clone, download, test it out, and then push changes if you want to contribute to it. The manual and the API reference are in Read the Docs. We are continuously improving the docs, so if you also have suggestions for it, feel free to do it. And the manuscript is in bioRxiv and soon on Nature Biotechnology. With this, I would like to thank the entire team, Matthias, Alberto, and Alara. It was amazing to work with these guys, such talented researchers, and all the funding agencies, of course, and all of you today here. Thank you.
Fantastic. Thank you so much for this super interesting presentation. And again, I think both the amount of data and data sources you have integrated and the types of analysis you can run are super interesting. And obviously, most interesting for the community is that this is open source and reusable. And let me have a look at the questions. So Umberto is asking, how do you handle multi-target drugs in your target prioritization?
That's a very good question. In this case, we were lucky. We had almost one-to-one relationships, but multi-drugs, you could find other kinds of data to prioritize them. We use diverse reactions. For example, you could use higher scorings, lower scorings, different kinds of evidence where that association came from. It all depends on what you prioritize.
Mm-hmm. So I think that that already answers the follow-up question. Do you account for druggability scores in any way?
Druggability scores, if it's in the drug bank database, yes.
Mm-hmm. Right. And another question about open source and accessibility. So is it possible to run a local copy of your system, or how is it possible to reuse your code and your project?
So you can, all of these notebooks, they are recipe notebooks, so they are on the GitHub repository. If you clone or download it, you can install. We have made the installation a bit more simple. You can run a Docker image, for example, and then you can just open it in a Jupyter notebook, and you have everything I showed in this example will be in this notebook.
Mm-hmm. Fantastic. So it's really easy to reuse and test. I think that's fantastic that people start wrapping their stuff in Docker containers and so on.
If you are using the Jupyter notebooks, you just need to have a little bit of knowledge on Python. That's why we also made the automated analysis pipeline without people having to code in anything. But if you do have some knowledge, you can just manipulate at your will the data and mine the database in the notebook.
Mm-hmm. Okay, so thank you so much for your presentation. I think it was really interesting. We got a lot of feedback, and I'm pretty sure that a lot of people will be interested in trying the clinical knowledge graph. And good luck with the publication, of course. So thank you so much. And with that, it's a pleasure to announce the next speaker. Alexandra Jarasch from the German Center of Diabetes Research will present how they went from queries to algorithms and to advanced machine learning. Your stage, Alex.
Good morning, everyone. Thanks for joining my session, and thanks also to all the other speakers from yesterday and also Rita today. It's really fascinating what you guys are doing, and I always think that we are doing graphs, but everybody's doing graphs, so I'm pretty much amazed of all these applications. So how can I share my screen here? Share my screen. Here we go. All right, so you should see my screen now. I'm showing you today some applications that we are doing in the German Center for Diabetes Research. First of all, a little summary of what we are. We are a nonprofit organization and a federal institute in Germany here with 5 partners and several other associated partners, which sums up in roughly 450 researchers that are having expertise in basic research, but also in clinical trials and clinical research. On a kind of data level, we have a variety of data. Obviously, we have very unstructured data. It's very heterogeneous. Usually, it's not connected in a federated state like Germany, and so we term that unfair. So here's a overview of my team. So we have 4 developers and myself who are sitting in Munich and Germany, and our challenge is that we have data silos. They are scattered around Germany, and usually people are asking scientific questions that are usually very easy. How many data samples do we have from blood in a mouse or in human? It's a very easy question on the one hand, but to answer this question on a data level, it's not that easy because the data is scattered. And this is what we are tackling. We also have another challenge because we have many different users here. For example, we have a scientist that is usually not trained to be a computer scientist, coming from the clinic, coming from as a biochemist. So they have questions very from the scientific level or from the medical level. But we also have data scientists that ask query questions or query-like questions. And in the end, we want to make access to them to all the same database. Why are we using graphs? I think you're familiar with questions like these. Are human type 2 diabetic genes enzymes acting on metabolites, which in turn are regulated in the pig model? Right, this is a usual question that a researcher would ask. What does it mean on a data level? Basically, it means is A connected to R? And I'm showing you here one Excel sheet, and I give you 3 seconds if you see if A is connected to R and in terms of a data point. It's 21, 22, 23. And I guess you didn't see it. I'm asking you the same question now. Is A connected to R when we transfer our data into a knowledge graph or into a graph database in general? And I think you can easily see that A is connected to R. That means in the end, we have basically a graph problem, right? So we only have to transfer our relational data or our Excel sheet data into a graph so that we can make scientific questions. I think I don't have to show you why we use graphs here. Graph is basically everything what we have in biology. Everything is connected. Everything is dependent on each other. We use Neo4j because it's easy to model. It's extremely flexible. Also easy to adapt. So when we have to reshape the graph, it is very, very easy when we compare that to usual SQL models. I think we have quite a huge graph database now, and so we are still scaling on one server. Many of our queries are cyclic dependencies, so that is very, very easy to answer. And last but not least, we're using Graph Data Science Library and graph embeddings. I will show you that in a second, and that's why we're using Neo4j. Diabetes is a connected disease and is connected also to other indications like cardiovascular diseases like stroke and heart attack. Diabetic patients get Alzheimer, diabetic patients get even liver cancer, infectious diseases when we think about COVID, also lung diseases. So that's why I see diabetes as a connected disease in a connected network. And that's why we have to see not only diabetes data, but also connect other data from other indications. Well, the concept is pretty simple in our case. In the center, we have a knowledge graph, which we build up from public data. We connect this by using natural language processing and inferring knowledge. This is the second layer, doing named entity recognition. And in the end, this is the outer layer, we connect our DCD data to the knowledge graph so that we can ask scientific questions. Let me talk about a little bit about the stats. So currently we have a production server with roughly 330 million nodes and roughly 1 billion edges. On our hard drive, which is only SSDs, we have roughly 500 gigabytes. We have another developer server, which is way larger. We all run that on a single server machine, 60 cores, 256 gigabytes of RAM. So nothing really special, I would say. No cloud instance. So we are running Neo4j Enterprise because of live backups and GDS library. We have some applications running on top of our database. I will show you that in a second. And we use the visualization, the interactive browsing by SemSpect, which I'll also show you in a second. Some words to the data integration. Most of our data comes from SQL databases and also spreadsheets. And here we use Python Neo and GraphQL packages, so Python basically. We have a Docker orchestration pipeline, which is basically open source, and you can also use that. We integrate data and then we enrich data, normally using text matching algorithms, named entity recognition, natural language processing, you name it. And I'm only focusing here on this example. We integrate genes and RNAs and proteins. I guess you're familiar with that. So genes are coding for RNAs, the RNA is coding for protein. And this is where we do inferring knowledge, which is in this case not inferring knowledge, it's like straightforward. We are creating this red relationship here, codes that a gene is coding for a protein in the end, in order to save some hops in the graph database. And this is what we're doing many, many times in our database. Here you see the overview of our data model. You don't have to read that here. I'm showing you that in a little zoom. Just to show you here the domains that we have. In yellow, we have literature. In orange, we have all the mesh terms and the keywords from the literature, also affiliations and scientists. The pink part are all the molecular modalities like genes and proteins, metabolites, gene variants like SNPs or SNP interactions, also chemicals. We integrated some ontologies in green here, disease ontology, gene ontology, phenotype ontology, and we recently integrated also gene expression data here in blue. Well, that's the zoom here on this data model. You see that the nodes are pretty much connected to each other. We also have relationships pointing on the same node, meaning that we have synonyms and mapping identifiers from different databases. So we also cover that. Let me say a few words of our challenge about the users that we have and the kind of input they need and output they need. So the challenge is, for example, we have a biochemist or a biologist. They do multi-omics experiments and they want to know something about the output. And this is where we have a specific input and a specific output. For this, we developed a Flask app where you can enter your multi-omics experiments, can be transcriptomics, proteomics. We have a pilot for lipidomics even, and the result here in this case is a very specific output. Where is the gene mentioned, in which publication, which kind of mesh terms, which kind of ontology is used, and so on. Then we have a second group of users, which are usually our medical doctors in the clinic. They don't really know where they want to start their research and they don't know where to end. So they are very, very freely to explore this network. And for this, we integrated a tool called SemSpect by the Rival company here in Germany. On the left side, you see something like Neo4j browser. You see all our node labels here, and you can drag and drop them easily into the big field here and do your exploratory search here. And all the nodes that you usually have and that are wobbling around in Neo4j browser are summarized here in very nicely meta nodes, so you can easily cluster them, you can filter them accordingly, and it is very, very handy to use that. The third user is the data scientist itself. So we have people who can read and write Cypher queries, Python code, and so on. So we have Neo4j browser as well as Python interfaces. So you can also access our graph using Python or R. Now, let me show you some use cases that we have in the German Center for Diabetes Research. And we go from very simple to more advanced queries. First is we want to query a friend of a friend. So we want to have a gene that is connected to its synonyms, but also to its mapped identifiers from other databases. And this is very easy here because we are defining a path. You see the Cypher query. We query a gene called TCF7L2, which is a typical diabetes-related gene, and we are mapping 1 or 2 or 3 hops, here in this case 2 hops, to another gene, which is a synonym or a mapping identifier. And this is like 0.1 seconds, and you get all the synonyms and all the mapped identifiers from the same gene. So what you see here in this graph is basically we're talking about the same gene, and we have very, very different gene identifiers. Second example is that, let's say we want to query that gene, and we want to find information that is connected to this gene. And this is where we integrated, as I said, gene variant data, so SNPs or mutations that are associated to diabetes. And we were interested in what can we find in these databases according to this gene. When we start from the very left, this is the type 2 diabetes trait or the phenotype that we have, which is associated here in cyan to a gene variant. This gene variant is associated to TCF7L2, the gene, as I showed you in the last slide, which have an internal identifier in ensemble 6934. And this identifier is coding for a transcript, so an RNA in blue, and this is coding for a protein in green. And this protein has basically a function, right? And this function is in gene ontology. So we have the function in orange and the ontology term or the ontology itself in green. And that should only show you here, all the colors are representing single data silos that we are now connecting natively in our graph database. The third example is using GDS library, and I will skip that slide because Martin showed that already. And I'm showing you here the example for COVID-19, where we could identify ACE2 as the most prominent gene using Google PageRank algorithm. Unfortunately, I cannot show you here our PageRank algorithm results from the DCD because this is still confidential. So we'll skip that example. Another use case is that we use now graph embeddings or node embeddings to sub-phenotype our diabetic patients. Now, let me explain how we do that. In the DCD, we not only have a knowledge graph with public knowledge, but we also have scientific data from basic research, but also clinical data here from our patients. And on the left side, you see there's very relational data. We have Excel sheets where they keep their data or some SQL databases, and we transform that now into a graph here in a very centric. In red, we have the patient who has a medication. We measured some biomarkers, has a type of diabetes, maybe has some surgeries, and so on and so forth. And then we are performing, in our clinical studies, we are performing experiments here in orange. And these experiments, we acquire parameters in green on the very right. And sometimes we have continuous time data, so we also keep track of the time here. So we integrate the data, and now we use molecular fingerprints like lipidomics fingerprints. On the left side, again, you see the Excel sheets where we measure specific lipids here. In our case, we have a platform that can specifically measure 116 specific lipids, and we represent this in a graph. Same is true for transcriptomics experiments. Here we have chips which have roughly 60,000 transcripts, and we connect them in Neo4j, also with the values of the different patients. And what we then do is we calculate fast random projections. So and in memory graph where we have node embeddings, we take the top 50 parameters, and we represent our patients with their molecular fingerprints in a vector so that we can do machine learning. And that's what we do. We then apply here k-nearest neighbor clustering with k equals 5, because we have in research we have roughly 5 different subtypes of diabetes, and so we can cluster our patients and can then learn more about the different subgroups, if they are responding to a specific drug, if they are responding to a specific treatment or prevention strategy, or if they not respond to it. In the end, we connect that to our knowledge graph, so that you can easily ask questions like, I have here this specific transcript on my chip. What is known about this transcript? Is that coding for this and that gene? Who's publishing about this gene? What is it connected to? Is there another indication like cancer or Alzheimer's? So we can easily query that. We can also query from the other side, coming from the right. We see an ontology term or a disease term, and we can query through our graph to our experimental data and ask the question, do we have data in-house to this and that event? With this, I'm at the end of my talk. I hope I could show you that we have quite a huge knowledge graph, which is our single point of truth. We connect in-house data, but also public data. We also connect cross-species data. So we have integrated animal models like from pigs and from mice, where we can do studies in animal models and then infer the knowledge to the human. I showed you some simple and more advanced use cases, and I showed you here the specific applications that we are running, so like the Flask app, the SemSpec, and browser. And with this, I'm already at the end of my talk. Oh yeah, that's the last comment here. We could increase our performance in research or in data lookup between 20 and 12,000 fold. So I think this is quite a remarkable number. So depending on your input, sometimes we are 20 times faster. But in our last use case that we had, we were 12,000 times faster than the normal approach before. So with this, I'm at the end of my talk, and I'm happy to take questions. Thank you for your attention, and yeah, I'm really looking forward to the other talks. And if you have any questions, let me know.
Alex, fantastic. Thank you. As usual, impressive to see your knowledge graph and to see kind of the amount of data you have integrated there. Let's have a look at the question section and at the Q&A. So there are a couple of questions, starting with Lisandra from Lewandowski, who presented yesterday. And the question is, which genetics database did you use for getting the SNPs and SNP interactions?
Basically, we integrate or we can continuously integrate GWAS data. So we download the GWAS catalog. I think currently we have version 1.0.4. And in this catalog, there's also the gene interactions, but also the SNP interactions. So sometimes we have a SNP interaction, like one SNP is depending on the other, or we have a cross inference like that. I don't know the correct term for that. So this is coming from GWAS data.
Mm-hmm. Okay, there's another question. Hi, Alex. From SemSpec, can you select data sets and run more sophisticated analyses?
Depending on what you integrated so far, if you integrate your data into nodes and relationships with specific measurements, I think you can do that. Shouldn't be a problem. Currently, we are not using that with SemSpec, but in general, I don't see any problem with that.
And I think what you can do is you can always export CSV files, right?
Exactly.
So if you have some filters and everything, might be worth pointing out that Health Echo has a public SemSpec instance for COVID graph. If you're interested in trying it out, maybe I can post the link in the chat a bit later.
Mm-hmm.
Let me jump back to the chat and Q&A and see if we have more questions. While I look for that in the various different channels, I would have a question. So, I mean, Rita and the clinical knowledge graph have also like a lot of very interesting data integrated already. And they have their own kind of data loading pipeline. You also have lots of data and a fantastic data loading pipeline, but there's a lot of overlap. So could you comment maybe, what do you think, how would you approach, sharing resources here? And integrating this or combining this?
I thought about that many times, seeing all this nice graph use cases from other groups, and I would like to reuse, like a data source or some knowledge that they have, and I think one approach could be like exporting or dumping a subgraph of the one graph and then importing that into our graph or the other way around. If we're not talking about the pipeline itself. But I think sharing subgraphs with each other would be a very interesting approach, so that data can be loaded in this or that way, but we can share that on a let's say on a higher level or on a database level.
Mm-hmm. So the idea would be to share it in kind of its final form, knowledge graph subsets.
Well, that would be one option, right? To dump it and to import that in your own database. But I also see a possibility just to reuse, since most of the part that we do is open source, to reuse that the Python scripts or other script languages, just to run that in another instance. Yeah.
Mm-hmm. Okay. So let me have another look at the chat. There are a couple of questions around accessibility. So can you post the GitHub link and so on? So you mentioned that your code and also the data loading pipeline are open source. How can people access that?
Yes, so we have to put that into our presentation. So when we share our presentation, I will give you a link to our GitLab repository so that you can reuse that.
Mm-hmm. Perfect. And let me have another look. So there's a bit of chatter on the chat. So there's a follow-up about the SemSpec question. Okay, thanks. I think it would be great if you could use tools like SemSpec to select sets for collecting data and then move to, for example, a Jupyter notebook for the analysis. So it looks like this is interesting to people in the chat.
Yeah, as Martin mentioned, I think this is pretty easy. So you can explore your graph and then export that into a CSV file and just import that into your Jupyter notebook.
Mm-hmm. Okay. So a last look at the chat. If anyone has another question, it's your chance to post it now. If there's nothing else, Alex, thank you for your presentation. Again, as usual, impressive knowledge graph, impressive data, and I think very interesting applications from simple to complex. I like that. So thank you so much. And I think with that, we can jump to our next speaker.
Thanks for having me.
Thank you so much. Let's jump to our next speaker. Just a second. There we go. So the next talk is from Xixing Wang from MGI, who's joining us from China today. And he's going to talk about Neo4j for bacterial genomes. Xixing, the stage is yours. I'm really looking forward to your presentation.
Okay, thank you, Martin, to speak in this conference. Hello, everyone. Nice warm welcome from China. Today, I'm going to talk about Neo4j for bacterial genomes. The talk's overview is simple. In contrast to the talks from the previous day, this talk is about doing small projects. The data are from public database and whose size are typically smaller than 1 GB. So all the analysis can be done on a local computer. In bioinformatics, bacterial genome analysis, we typically look at 3 different levels: gene level, genome level, and phenotype levels. Here, I'm going to show you that Neo4j can cover all these 3 levels. So I'm going to show 3 examples: Neo4j for genome analysis, for carbohydrate active enzymes with CASI, and for antibiotic resistance in the CARB database. So first about me, I am a bioinformatician who studied and worked in Bremen and Braunschweig. Currently, I am employed in Shenzhen MGI. My contact with Neo4j was short because my first project with this software platform was in 2019, but it was love at first sight. Since then, I use Neo4j for my knowledge management as a genome browser and also a backend database for many of my projects. And I also passionately writing about Neo4j in medium.com. So first off, genomes, they are different from just a bag of words because genes have structures. They are organized much like in the way of the natural languages. So they have orders. Traditionally, we save this gene information in a relational database, which is very heavy and they are very difficult to understand. But using a graph database like Neo4j, we can model this gene structure very effectively. So many biologists may be familiar with the file, which is used to save the genome information. EMBO file is a big text file. It is very difficult to read, to search, and so on. With a small import script, however, we can import all this information into a Neo4j database. And it's very easy to visualize these genes in the Neo4j browser, as you can see on the right. So the red node represents the organism, and the green node represents the content of inside this organism. And then each blue nodes represent one gene. And we can save many information in these nodes. For example, the genome size, the annotation, and so on. It's more than just beautiful pictures, because with these data structures, we can do a lot of things. For example, gene cluster analyze. Say you have a gene in mind, for example, GH16 from CASI, which degrades many agar polysaccharides. And you want to see the neighbors of this gene. So in Neo4j, if you save the genome data in Neo4j, it will be quite easy with just one query, as you can see here in the example. In this case, we not only find the 11 genes we are interested in, because Neo4j is going to pull all the copies of genes GH16 together. So we have unexpected findings here. We can find some super gene clusters that was previously not expected. Also, it is quite easy to edit the genome information in Neo4j. Previously, we used software like Artemis to edit the genomes. But with Neo4j add-on like Neo4j Commander, we can edit the gene annotation also inside Neo4j with a graphical interface, which is very nice. So that was the gene level. What about genome? On the genome level, we are no longer interested in the order of the genes. We are more interested in how many of each genes. So in this case, we have a genome matrix. So it is a very simple numeric representation of the gene contents of each genome. Typically, we are performing the so-called gene annotation analysis. For example, in this case, we have two genomes, the blue one and the ochre one. Because both of these genomes, they will share some genes and they also have some unique genes. Together, the genomes is called the Pan-genomes. We are going to look at each part of these genomes so that we can know the relationships between these genomes. So this is typically called the autolog analysis. And in Neo4j, we can do this kind of analysis effectively. There is a unique challenge in the in biology analysis in this case, because things in biologies, they are in orthologies. For example, in taxonomy, we have phylum, class, order, and genome. Also, we have orthology in gene annotation. One of the most heavily used orthology, it is the CAC gene annotation, as you can see here in the right example. So metabolism, carbohydrate metabolism, and so on. As you can see here, we already can see the graph structure here because we can see nodes, we can see collections. So we can represent all this orthology together very effectively in our genome analysis. So in this concrete example, I'm showing you that how I'm going to represent a genome in Neo4j. So on the left side, in the red nodes, you can see I can represent the taxonomy with the Hess taxon relations. And then on the right side, I can test the Hess-KO relations that summarize all the gene annotation of the particular genome. So in just one schema, I can have both the taxonomy and the gene annotation in one place. And with all this information in one place, we have great advantages. Let's say we want to do one small analysis about a bacterium called Comobacterium ATCC 53434. There are in total 6 such Comobacterium in the database, but I'm only interested in this particular ATCC strain, and I want to see what genes are unique in this genome. They are not shared by the other 5. So in Neo4j, we can use the cipher effectively to do this. As you can see in the screen, the first 3 lines is about creating a gene filter. So basically, I just exclude our ATCC, and then I will combine all the genes in the other 5, and then I will collect the result using the with clause. And then the last 3 lines, I will list all the genes in my ATCC strings and then compare them against the filter. The results are the unique genes in our ATCC strings. And then with these results, we can look at patterns and see if there are any interesting things. And in this case, yes, we can find that there are many genes that are related to iron transport proteins. For example, I can find 2 groups of siderophore proteins, and then the last one, it is a complex of a ferric hydroxamate transport, and also one more gene, the ferric iron reductase protein FUF. So you can see, as you can see, with a very simple query, we can discover something new that is not mentioned in the literature. Also, we can do phylogeny. Normally in bioinformatic analysis, the phylogeny is normally carried out based on the 16S or other protein ontologies. In our case, we can extend this analysis with Neo4j, and then also to do a very small phylogeny very quickly, just by looking at how many shared KOs are there between different genomes. Again, taking our ATCC string as an example, with these 3 lines of cipher query, we can quickly calculate the shared numbers of KOs between the 6 strings. And as you can see here, the C. vaccini has more than 1,800 share KO, so it should be a very close relative to our ATC strings, while the last one, the SP257-1, is a remote relatives. So we can compare this result to the 16S to see whether these two phylogenies are congruent. We can extend the same the process to other domains too. For example, in carbohydrate degradations, the CASI database collect a lot of carbohydrate-related enzymes. So we can look at the so-called carcinomes, whether a particular bacteria can degrade a certain kind of polysaccharides. We can use Neo4j for this purpose too. In this example, I show you the reannotation of a bacterial genome of Formosa agariphylla. This genome was published more than 10 years ago, but since then, we have a lot of new annotations. So as you can see here in the graph, the central node represents the genome, while the orange nodes represent the CASI annotation, these genome processes. And just as the same procedure as the CAC, here I calculated the unique CASI annotation of this genome. So only for Formosa agariphylla possess, but not his sister genomes. And as you can see, we can also find 3 groups of genes that have particular biological significance. The first group degrade 1, the second group degrade pectin, and the last one degrade sulfated polysaccharides. And they are all cellular components of the green algae. So again, this new annotation confirms the agarolytic lifestyle of agariphylla. It is more than that. We can extend this knowledge to other polysaccharides too, not only green algae, but also cellulose. As you know, cellulose play a great role, big role in the biofuel and other industrial domains. So it will of great interest for us to predict which bacteria can degrade cellulose. However, among all the CASI annotated genomes, only a small part, like you can see here in the graph on the left-hand side, only a small part of the genomes have been experimentally validated against cellulose degradation. So the blue dot. While the vast majority of the genomes, we have annotation, CASI annotation, but we do not know whether they can degrade cellulose. So this project is to use this small subset of genomes to train a graph model and then use that model to predict the cellulose degradability of the whole R data. So in this case, I use Neo4j's graph data science library. And first, I embed the graph information in 5 dimensions in the embedding step. And then afterwards, I use the Neo4j cross-validation procedure to train a logistic regression model. As you can see, the result here, it is the training score of 0.67 and the testing score of 0.65. At first glance, the score were low, but however, I was optimistic because I have carried out the whole R prediction too, and I use this model, to predict which genomes in the whole R datasets can degrade cellulose, and the prediction returned 11 positive cellulose degraders. And then I look at the literature to find confirmations. Only 5 of the genomes have been experimentally tested against cellulose. 3 of them have been confirmed positively. The Streptomyces placinus, it was listed in BacDive as an undetermined against cellulose. So this is a wildcard. However, the second example here, the Mycomonospora HM134, is a strange case because this genome contains all the classes of a cellulose degrader and more. However, the experimenter said this genome cannot, this bacterium cannot degrade cellulose. So this it has a big question mark for me too. So also in this project, I have implemented the GraphQLs. So because not many colleagues in my group can use Cypher. It is a new language. However, most of them can use GraphQL because the syntax is really easy. As we store all the genome information in Neo4j, we need to also provide an access for all the colleagues, the non-Neo4j users. So the GraphQL provides a very effective way to interface all these users. And because GraphQL do not over provide data, so it saves bandwidth and it is very efficient for data loading. So finally, I'm going to present one more example. It is about the antibiotic resistance. As you know, the human society has overused antibiotics, and that leads to a widespread antibiotic resistance. It's an emerging global disaster because it is predicted in 2050, more people will be killed by the antibiotic resistance, and is going to make a lot of medical procedures impossible. For example, operations or something as simple as device replacement. So it is very urgent for us to deal with this situation. And the CARD database is dedicated to the antibiotic resistance. In this project, I downloaded the data from the CARD database and then imported them into Neo4j. The schema, as you can see here, it is simple. So the lila nodes represent bacterial, and the orange node represent mechanisms. And then the green node represent the antibiotics. So the green nodes conveyed antibiotic resistance against the antibiotics. So the bacterium process the green nodes, then they can resist the corresponding antibiotics. Here you see two examples. On the left is the fluoroquinolone resistance, and on the right is the cephalosporin resistance. But even though they are widely resisted by many bacteria across gram-positive and negative, but the situation and the landscapes are different. The fluoroquinolone resistance is like a 9-head hydra. So if you want to completely eliminate this resistance, you need to cut off all these resistance mechanisms to make that work. However, the situation is different for cephalosporin because it seems there are only 4 mechanisms shared by many bacterium to evade the effect of cephalosporin. So in theory, we just need to plug all these 4 holes to eliminate this resistance. So we can see with a simple analysis like this, we can see how we should handle different antibiotic resistance in practice. So after these 3 short examples, I hope I can give you the impression that Neo4j can serve as an all-in-one genome browser, as a biodata warehouse, and also as a data mining tool. In comparison to relational database and SQL, it can deliver insights more quickly, in my opinion. It also use machine learning from the graph data science library, and it can predict new connections. In our case, for example, it can make new annotations or the missing annotations, and it can predict new properties. For example, in our CASI example, it will be the phenotypes. And also, we can set up GraphQLs as an interface for non-Neo4j users. So it is a database for effectively everybody. So with this slide, I want to conclude my talk, and I want to thank the following people. My tutor, Hanno Tailing in MPI Bremen, and the Neo4j community, because they have welcomed me warmly and provided me with ample support in my journey. And I also want to thank my current employer in MGI, because they have supported me in my research in the graph theory and the graph database. And I thank you all for your attentions.
Fantastic. Thank you so much for your presentation. I think you showed some really interesting concepts here. I will jump over the Q&A section and to the chat to see if we have questions. And while I do that, I would like to ask the first one. So what do you think your data model and your, let's say, analysis concepts and how you model annotations, do you see any limitations in using the same models or transporting the same models to higher organisms, human and mouse?
At the moment, I see there are several. First, because this annotation is really rich in relation-rich data. So if we are just doing single genome analysis, currently I cannot think about many scenarios for this this graph database model to use it, because currently you can see in my cases many of my examples are about comparisons. So comparative genomics, that is what I want to emphasize. So if on the higher scale eukaryotic genomes, I think you can compare different genomes, but at the moment, it is still on the gene level. So I don't know if you can do it on a SNP basis. So if you have single nucleotide polymorphism, can you still use it? But maybe it is a very interesting area I can explore. So can we go deeper than the gene level, as I show in this slide? So can we do SNPs, mutations, and yeah, and maybe also on the higher up level, not only phenotypes but population genomics, and so on. So yeah, can we show the overlaps between different populations?
Mm-hmm. Yeah, I think that would be really interesting. Let's go to the first question from the chat. And this is a question that we had a couple of times yesterday, and it seems to be interesting for a lot of people. So do you have new data frequently, and how do you handle updates?
Right now, all these 3 projects, they download the data from the public database. For genome analysis from CAC, the CASI is from CASI, and the antibiotic resistance is from CARP. So my update schedule is completely dependent on these 3 databases. As long as the database have an update and the curators have the need, we can update it. Currently, there is no, let's say, an automatic way to just stream feed the data, and yeah, it is like a snapshot of these databases. However, I can see the potential that we can add our own data. For example, if we have new genomes from our sequencing, we can add our genomes in all these examples. So we can combine the public and the private data. So we can do, we can crystallize some new knowledge from our private genomes, with all this public knowledge. So you can compare your new genome with the known sister genomes, like in the first example. So you can have all the benefits of the public data too. So, but yes, currently there is no like a live streaming mechanism, because these databases they do not kind of provide me like a feed or something, but they provide APIs. Like CAC, they provide APIs, so we can regularly update the data with their APIs.
Yeah, I find this question very interesting, because we had it after I think every talk yesterday, and not from the same person, always from someone else, because I think in terms of data model and data processing, updating is a very interesting challenge. But at the same time, we know that the knowledge out there changes. It's not static, so we know that we have to update. There's a bit of a follow-up to that question with the updates. How do you handle new data in terms of machine learning results? Do you have to do the embeddings and other analyses again?
At the moment, my current understanding of this graph data science library, yes, this model needs to be trained again, because you can currently I do not find a way to increment the model, so I can not maybe do something like a batch thing. But I think as long as the graph data science library can provide us with this capabilities, we can do this. But also I want to mention at the moment that the data for all these 3 projects, they are small. So the training, it will be like lightning fast. It is not the issue of the training. I think it's more than the data cleaning and the concepting that takes the most of the time. So the training and the prediction will be just like 2 clicks away. But the model, yes, it is static at the moment.
Okay, so the model is not updated, but the performance of the GTS, at least on your data set, is good enough that you can just rerun it.
Yes.
Okay. Let's maybe talk a second about the data. So is your database publicly available? Did you share the database?
Yes, they are all publicly available. As I also have written all these 3 projects in my medium.com articles with all GitHub repositories with all the code and data. So and everybody is welcome to interact and to join me on this journey. So I also want some inputs from the broad communities.
Yeah, that's fantastic. So maybe you can join, you can share the Medium articles, blog posts with me, because I'll post them in the Big Marker chat so that everyone can have a look at the articles, at your description, because I think it's very comprehensive, also with a lot of figures and so on. And obviously the code is available, so that's really, really cool.
Yes.
Let me have a look at the chat and the Q&A section if we have something else here. Maybe one last question. I found it very interesting that you said that you use GraphQL to give everyone access because it's so easy to use. So yes. We had a couple of mentions of GraphQL yesterday, but I think there are still a lot of Neo4j users who don't use GraphQL APIs. So could you maybe comment a bit, how difficult was it for you to set up the GraphQL API? And how easy is it really for end users, for non-IT or let's say non-graph database users to use GraphQL?
Actually, the setup was quite easy. There is an excellent post in the Neo4j forum, and I followed that post. And then I also have written my whole experience in the same article in the medium.com. So the setup of GraphQL was relatively smooth. Everything from there, I think. It depends on how we model the data in the GraphQL, because so in my case, I have, for example, modeled the whole data as I show in this graph. So my colleague can pull the taxonomy and the annotations from the GraphQL as they can see in here. So in the mental model, so as long as they understand this graph, this graphic, they can pull data relatively easy with my GraphQL. And the usability of GraphQL is quite nice because it can be accessed with different languages, not only this interface, but also they can pull the data from their R, because many of my biologist colleagues, they use R and Python. So they don't need to stick around with this localhost 4000. They can directly pull the data without knowing Cypher and to do their own analysis. And for those graph folks, they can keep doing their Cypher and doing amazing stuff. However, for the non-Neo4j user, they can still have their access to the same data. So I don't need to keep two different annotation databases. And there are even talks in the group that they want to change all their data storage into a graph database. And of course, if we propose that, many people will protest. However, if we say, don't worry, we have GraphQL, and then people, okay, that we can live with that because they know how to pull data from GraphQL. So I think it is like a nice facade for many of our users who still have not learned Neo4j. But for those Neo4j lovers, they can use their Cypher as usual. So I think they can live both nicely together, and we provide them both so that we can just keep one database, but all the users can use it.
Yeah, thank you so much. So I think your blog post also on the GraphQL bit will be a very interesting resource for the community. So thank you so much for your presentation. Thank you so much for being here. It would be great if you would share the links again.
Yes.
And so thank you so much. And I think with that, we can move over to our next speaker. The next talk will be about modeling multi-omics data in Neo4j to identify targets for strain development. Piyush Sahu, bioinformaticist and data scientist at Bacterial, will present this project. So the stage is yours. Looking forward to your presentation. I think you are muted, so we can't hear you.
Okay. Can you hear me now?
Yes.
Okay, good. So thank you, Martin, first of all, for the invitation for this talk, and really, to be able to attend this workshop where people are working, with Neo4j and the graph database in a very different dimensions, and also actually in a quite overlapping manner. So today, let me share my screen. Just let me know if you can see the screen slideshow.
We can see the screen and the slideshow.
And a proper slideshow means in the full screen.
We don't see that yet. We still see the PowerPoint window.
let me then try to correct that. Oh, I have to share complete screen in that case. Oh yeah, that makes sense.
Perfect. And now we can see the slideshow in full screen.
Okay, that sounds good. Okay, so today what I'm going to talk about, which actually our preceding presenters have already touched upon and actually explained in detail their use cases. So in Clariant, just a little bit about Clariant. So Clariant is a specialty chemical company. However, the biotechnology unit of Clariant is working towards bioethanol production plants, and so we are setting up the process to convert cellulosic or hemicellulosic material, which is also referred as 2G materials, to bioethanol. And in this whole process, there are strain development, which is a very big focus of the research. And in that area, because you need to identify what is good and what could be what could make a strain better with respect to certain production of enzymes and other things. So we came up with this. We always come up with the questions from the scientists that how to make this process easier, identifying something which is good for the process. Okay, so let's. Okay, so this picture is quite actually nice for us, and when we think about how data actually comes together, or how data can make sense, it is very interesting, because when you look at this picture, it shows you that that how perceived observation could be misleading in the absence of a complete overview. And this is very true, I think, with respect to multi-omics, or actually a lot of biological data, because without having all of its parts, observation derived from a single technology in either proteomics, transcriptomics, metabolomics, any, could be misleading. So our aim was to develop a platform where we could utilize, or actually put all the data together, and then ask questions which can result, or give us meaningful results in that case. So overall aim is to drive the discovery, so the scientists can really identify what is the reason for a certain phenotype if we are looking at something. Okay, so what we have here in Clariant, what we are working on is, we call it the multi-omics platform. So it started with combining the multi-omics data together. However, it has grown further now, and we are incorporating not only omics data, but also phenotypic data, assay data, and other data types. But to go ahead with the name, this multi-omics platform can be divided into two parts in that case. So the first part is, of course, data storage in a knowledge graph, and then the second part is visualization and analytics, because in the end, once the data is in, you need to find a clever way to visualize and make sense of it. So coming to the first part, so the data storage. So we had certain requirements when we were looking into how data could be stored, because we are not talking about only storing data for machine learning, but it is a data storage platform for us. So the requirements for it, so we wanted to store metadata for each experiment, and all the samples in this metadata. It could be process data from fermenters. It could be at different time points. It could be any sort of comments which are important for scientists to infer the results or make some conclusion at the later time point. Then we have, of course, all the different omics technology and phenotypic data which need to be integrated in the platform. We also wanted to have cross data analysis, so where genomics, transcriptomics, and proteomics and all the data could be kind of extracted together and could talk to each other. Another very important aspect for us was to store multi-organism in single database, and also actually multiple genomes in one database. So when I say multiple genome, means multiple versions, because we work with many organisms which are not that well annotated. So we do evolve the organism genome annotations, and then it has to the model has to somehow make sure that all the data is there and we can access it if we want to. Of course, another very big part of knowledge graph is to enrich this whole data with available knowledge, expert annotation, and as much as information you can find about omics in terms of gene, protein, transcripts. And so, and of course, so like I said, so in the end, of course, everything has to be connected in a biological manner, so it makes sense, and a fast extraction of the whole data. So these are, of course, the requirements. And with the Neo4j, I just wanted to show you a very small, very abstract view how we want to, how we think that it should be done. So you have metadata, which is connected to captured data, which is connected to biological entities and the annotation. And now going a little bit into detail, so what I will do, so I want to share with you that how we think our data model works at this point. Means, okay, it took quite a lot of revisions. I think we are already at 8 or 9 revisions of the data model, where we had to change quite big parts of the data modeling. So in this slide, we have 2 parts, or we are talking about 2 parts of the data model. First, here we have the metadata part of the data model and the captured data part of the data model. So like I said, the metadata is a very important part for us, because we are storing process data and we utilize those data. And just to say, so it is evolving still. So what we have here, so we are storing data from the experiment side. So there are multiple experiments done in a lab, and these experiments have metadata about them, what are the conditions, and other data which could be attached here. And of course, this experiment could be chain itself if it is derived from different experiments. And then, of course, experiments results into samples, and these samples could be transcriptomic samples, genomic samples, or proteomic samples, or phenotypic related sample assay data. What we wanted to do, we also wanted to capture, if we want to group these samples into some meaningful manner, if there are some conditions which we want to use to group them together. So we used a non-chain event. So we used a sample group. We call it a sample group here. So you can easily put them multiple samples from different experiments together. So they can be easily extractable if need comes in the future. Now coming to the capture data part, actually. So this is where our experimental data lives. Of course, so the first 2 nodes, measurement and analysis, these are nodes which store a little bit more about the metadata of the capture data, something like what type of technology it was in terms of what measurement it is from. So something like here you have variant data. So where we are talking about whether there was a single nucleotide polymorphism or insertion deletion, or even structural variants could be modeled. And then with the similar data model, one can also store transcriptomics estimate, and we can further extend this to differential expression, because we from the transcriptomics estimate, you derive differential expression. So what is very good, and what we think it's quite clever here, that you can combine actually different analysis node to create a new analysis, which can result into a different result or different data point. And similarly, we have other data types such as phenotypic data, which can be stored. So what is very, very interesting for us in this point, that the overall data model is same for all the different data types. So when you are querying it, you can utilize the same path with certain parameters, and you can extract actually all the different data in single queries. So this is about the capture data. Now coming to the later part is very interesting, and means we have heard a lot about how the knowledge is stored. So we are coming to more into the genetic background and how the biological entities and annotations are connected. So I will go ahead, because this is a complex topic, so it needs at least its own slide here. So what we want to, like I explained earlier, that we want to actually store different genomes, different genome versions, multiple organisms, different strains, and also actually if a strain has a vector inside it, we also want to store that information in. So if you are looking, if you look here, so we start our model from organism, where organism has multiple strains connected to it, and this organism also has multiple reference genomes. This reference genomes has genes, transcripts, protein, and of course, so a gene could have multiple transcripts or multiple proteins depending on the different splicing events or alternate exon selections. But what else we have is strain is connected to a different part of data model, which actually defines vectors. So where you can store data about if there are some external DNA which is introduced in your strain. This is a very powerful thing, because with this we get the flexibility to actually look into strains or even the non-native part of the genome. And coming to the second part, of course, so the annotation. So of course, genes and proteins, they have to be connected to what is known about them. And for this, we are going with annotation part of our data model, where we create additional nodes for different annotations. And so we have different source here. You can get annotations from UniProt, NCBI. You have you can also get EC numbers about enzyme functions. We have Go annotations there. And a good part, a very good thing is that what we thought, that annotations can actually connect to themselves and to the entities. Why to connect to themselves? Because if we want to store actually individual IDs to individual nodes, if two different IDs come from the same database, they are together. You can also mark that what kind of actually what do they actually associate with, whether gene, transcript, or protein. So this gives us more flexibility when we are getting this data out. What do we want to look at to give scientists and data scientists a way to filter their annotations for looking for functions? Okay, so this was about the data model. So we so after the data is in the in Neo4j, the next step is, of course, the extraction, analysis, and the visualization of this data. For this, what we did, so we use Python and Jupyter to create a user interface which allows not only data scientists to query the data and to make sense of the data, but also to the lab scientists. Because in the end, we want our scientists to be empowered, so they can look into a huge amount of data, or the data which they had produced 5 years ago, and just with certain couple of clicks, they can extract these data and compare with all the different processes which are they are currently performing. So how do we do it? This is just a schematic representation of how different parts are connected. So of course, like we already saw, so the omics data is already in the database. Biological relations are also present already in the Neo4j database. But in-house, of course, we have LIMS, electronic lab notebook system, where our all experiment data, registry, and inventory reside. And actually, there is a lot of important data which is required from this database, which could be ported to Neo4j or to our multi-omics platform to make more sense of the overall data. So in this process, like the schematic shows us, so we combine Neo4j together with Python and Jupyter to create these user interfaces, which can be used to query and actually analyze data. Due to data sensitivity, I am not able to show you a live view of how we play around with the data, but I would like to at least give you a feeling how the notebook and Python really empowers data extraction and data visualization. So this is one screenshot of the Jupyter notebook, where scientists can actually select what organism they want to look for, what reference genome they want to work with, and then they are taken to another step where they can select different or multiple projects together and different analyses together. Then they can select what different properties they want. So this particular example is for variants, where they want to visualize if what different mutations or different variants are present between different strains, between different projects, and what different information we can gather based on annotation for them. And of course, some other things where they can filter out their initial observations with certain technical something like germline mutations or intergenic, or they can select structural variants and things like that. So this is on the variant side. We similar have something for the transcript side, where they can combine variant data together with the transcript data. So there they can select which project they want to combine the variant data with, and different analyses they can select. And they can also select certain cluster from variant, which I will show what do I mean with it in the later slide. So so as I cannot show you because of the data sensitivity, the whole process, but what I would like to give you a small taste of what it could do, because saving our whole data on Neo4j, we are talking about the whole multi-omics array of the data. So we can extract them together in a very, very easy manner. So within a couple of clicks, we can get all the data out, and it can be visualized, and data processing is performed on it. So this is a heat map which actually has multiple data types together. So in the first row, you have mutation information about certain genes. You have differential expression between different analyses. You have even noise RNA expression if you want to further look into how the RNA expression was. And this heat map was already clustered, so it could give you some patterns with an unsupervised clustering manner. So this is actually an interactive heat map. So of course we cannot see here, but to go over it, there will be a pop-up window which shows what gene it is, what kind of mutation it was, or in this case, how the differential expression is there. There are also information about the function of the gene. So if the scientists, just in case, are interested in some cluster, they can further zoom into cluster, look at what different genes are mutated, what kind of effects they can have on the function. So this is one way of looking at data. Then further, we also we have connection to the phenotypic data and other part of the omics data, proteomics data. So in this figure, what we have, it's in case a fermentation was run, which resulted into certain measurements for certain substrates, like here can be shown on the top. So what you can see, there is some abundance for certain substrate within different samples. Each column is one sample. And then we can right away on the fly compare it together with the proteomics data, which is produced for the same fermentations. And then we can see easily, and of course, so we have in our user interface, and scientists can select different enzyme classes if they are interested, or they want to investigate what kind of enzymes could have differential effects in different processes, and if they can find different patterns of protein expression which could correlate to what we observe in the phenotype. So we have another pipeline which runs on top, which tries to identify correlation between phenotype and enzyme classes or different enzymes and proteins. So this all comes together in the end to give scientists a way to generate hypotheses, to identify important features in strain development, and many more, actually. So but this, I would like to just go ahead and give what we think. So we are currently, I would say, at the early point of our multi-omics platform development. However, we think we have achieved quite a lot with respect to connecting all the data together, extracting data in a meaningful manner, which could actually result into hypothesis generation and identification of specific features. But what are our future plans? So we would further like to integrate, of course, more information about annotations like metabolic pathway regulatory networks, expert annotations, maybe information from publications, because a lot of organisms we work with are not well annotated. So publications are a very important information source. Probably information from the patents and other things. We are also looking for a way to automated hypothesis generation, where scientists currently they have to look into data and try to identify patterns, but we are working towards something where a rule-based system can be in place, which can already give some hypothesis based on the data they have selected. And of course, machine learning and graph learning is one of the future goals for us, where we would like to use this data, this highly connected data, to further advance our strain optimization and other discoveries. So with this, I would like to acknowledge our team. Frank Walrap, he's our head of group computational biology. We work together with Martin and Jamie, who are consulting us on Neo4j and other technologies. We have constant inflow of interns. One of our interns who really helped us with development of Neo4j was Ashok Vardarajan. He has left us, and after that, we have many interns now who are currently, they have worked and will be working on these projects to make it take it further. With this, I would like to thank you all for your attention, and I'm open for questions. Thank you.
Fantastic. Thank you so much for a great presentation. I think it's just cool that we now kind of with the last two talks kind of closed the circle. So we now really covered everything from patient level data that we extract from OMOP down to variant annotation and microorganisms. So I think this is really cool. Before we jump to the questions, I would like to announce that in our closing session today at 1:30 CET, We will have a small panel discussion with speakers from yesterday and from today, where we want to focus on the idea of sharing knowledge and resources again. So just exchanging the first ideas, how can we share data models, reuse data models, and so on. And Piyush will join us for that. I think that's going to be very interesting because I think your multi-omics modeling is obviously very interesting for everyone in the community. So let me jump to the chat and the Q&A and see what we get here. While I do that, I would like to reuse the update question that we had after every talk. So how do you handle data updates and changing data sources?
That is indeed, I think, a question which is worth looking into always, because data update is always a big issue when resources which you are using are changing constantly. But I think it is kind of same for all the presenters, and I think probably for us as well, that you have to always track what different changes are happening. And of course, then you have to update either the data model, because if that is something which needs to be changed, the whole data model has to be updated. But in most of the cases, it is only the data which needs to be updated. However, when we are talking about the update itself for the data, I think that is also something we are currently working towards, where putting data into the data, already created data model should be automated, where we have an API or a user interface where you can easily throw in the data and it is sorted according to where it should be stored and updated. And of course, because when you are updating data, there is one question which we try to already tackle. So there has to be some sort of consistency tests which we do perform on our data after the uploading. So the data is checked thoroughly that there are no wrong connections present, or wrong data has input to wrong nodes. So this is something we are focusing on quite a lot, because we want to create or maintain data accuracy on that level.
Mm-hmm. Yeah, I think that's it's always an important topic. And again, this question came up a lot. And let me again have a look at the chat. So just as a side note, I posted the blog posts from the previous talk to the Big Market chat. Another question is maybe, and this is obviously also something that came up a lot. How do you take care of data quality? How do you make sure that certain relationships in your database, when you collect so many different data sets, are valid and true?
Yes. So indeed. So data quality is important, and because the whole Neo4j idea is connecting data, so wrong connections could lead to wrong conclusions. So for that, in-house, we have developed a package which expressly takes care of it. So because our data model is quite nicely defined, I think, so we do not have, at least up to now, loosely connected parts. So we can really make sure that the data is connected in a proper manner and in an automated way. So if the query is happening, because in the end we are giving this data to scientists who will extract the data, and they will take it on the word from us that this is the correct data they are extracting. So the data quality is, of course, of utmost priority here. And that is something we take care of, I think, quite well with the automated scripts and testing data regressively after each update.
Mm-hmm. Okay. So thank you so much for your presentation. Thank you so much for sharing your data model, and all your whole approach. And again, I'm really happy that you will come back in 1.5 hours after 3 more talks for our closing session and our discussion on how we can, use all this and reuse and share and collaborate. So thank you so much. And with that, I would like to hand over to our next speaker. David Moutin from Aarhus University will talk about unveiling the knowledge and knowledge graphs. And this is going to be a bit more science focused and data analysis and algorithm focused. So David, stage is yours. Really looking forward to your talk.
Thank you, Martin. Thank you for organizing and for inviting me here. It's really nice. So let me share my screen. Okay. Okay. I think it should be visible, right?
We can see your screen, yes.
Perfect. Okay. So thanks again for the invitation. So this talk would be on the algorithmic side. So some of the challenges that many of the companies might face, and I'm very happy also to take the conversation later on, also via email. So my name is David Moutin, and I'm an assistant professor at the computer science department at Aarhus University. And today I would like really to talk something, once you have the data, what can you do, or how can you really unveil the most powerful information behind your knowledge graph. At least one of the algorithms or a couple of algorithms are easily implementable in Neo4j, even though we didn't, because we basically play with some prototypes. Very briefly about me, so I was a postdoc at HPI in Berlin, and where I met Martin and I met Michael as well. And I got my PhD from Italy, and I worked with graph for a long time under different aspects. So for me, graphs is a very generic abstraction. So especially knowledge graphs, it can represent many different things. So like social networks, biological networks, or some sort of co-citation networks. So there are a huge wealth of information. They can be structured in a hierarchical manner. They have multiple different types, so you can embed different files or different formats, and they're very expressive. And this is what actually interested me from the beginning, because they can contain a lot of information which are hidden, which are not readily accessible by a query, for instance. So that's why I focus most of my time in working with graph mining, which is a very broad area which combines together multiple different disciplines. Data mining, of course, to find some patterns, some regularities. Databases to retrieve the information and to store the information in a proper way, and machine learning to start predicting. So what happens if you read a lot of this data and you can predict the existence of something that you haven't seen before. I want to point out basically, but very briefly, that my area goes in many different directions, and I have been investigating multiple different problems in graphs, such as alignment. So if you have two networks, how can I find, for instance, that two entities are the same? Similarities, I think it's very important, and I will touch upon briefly in this talk, but not so much into the details. I worked a lot with exploratory analysis, which really, I think it's a powerful tool that we should explore more, because most often scientists and the domain experts really need to find something in the data. And what is that something is not well defined. In many of these, in many of the disciplines, in many of the sciences, it's not very clear. We're trying to find something interesting, but what is this? It's up to definition. So, and of course, also, it was mentioned in the previous talk, cleaning is a process which is very important. So I'm trying to make everything automatic, scalable to large amount of data. And in terms of similarities, I have done a few works in this area, but they're mostly somehow to figure out in an automatic manner whether two entities can be similar. And this is a very broad question, a very generic question. In some cases, you really have to integrate domain expertise, so something about biology, something that you know more. But in some other cases, it is possible to extract quite a lot of information without knowing anything. And this is what we did. So knowing really nothing and not even having entities as in the proper way, but just having the graph structure. But okay, for today, I want to talk somehow in a very high level. So imagine that you have your own data, because I couldn't really fit to a specific types of data. So I just start with something that everyone can know. These are smart devices that currently can answer questions like what is the weather today, and of course it usually answers, okay, the weather is like that. They can be also personalized, of course. Another thing is where is Copenhagen located, for instance, and this is yet another question that can be answered quite fast by these devices. There are other sort of queries. There are sort of questions that are more exploratory, which is, can you recommend me a city like Copenhagen in Europe? This seems very, very similar to the previous one, but it's not, because here the user or any scientist here is asking something that, is there something similar about from this entity, from Copenhagen entity in Europe? And unfortunately, currently, the answer is unsatisfactory. So that most of these devices will not return the right answer. They will just tell you, okay, I found something in the internet querying in that way. But even the search engines cannot answer this. So ideally, in some foreseeable future, we would like a proper recommendation, but it's not an easy question here. We would like to have devices that answer like that. So Venice, Marseille, Hamburg, which are recommendations of possible cities. Or even better, that it starts a dialogue with us. So if we are scientists, if we are domain expert, we would like somehow to know, okay, is perhaps Venice something that you're looking for? And I believe that Venice is the right answer because of this and this reason. So somehow explanations also. The idea that how these queries works, so how this simple queries works. Well, besides of the fact of understanding the natural languages and understanding what is written here, behind there is a knowledge graph. So these devices operate on normal knowledge graph where nodes are entities, and these entities represent, for instance, Copenhagen or Denmark, and then you have some relationships among these entities. And this can be easily done with the Neo4j, with the cipher query, right? So you can just ask this and you will get the right answer. So once you have done the NLP work, then it's just a matter of having a good database behind. And the answer will come. But when it comes to this question, so to recommend some other cities like Copenhagen, well, the things becomes a bit more complicated, because the cipher query can still find the structure, but the answer is not there. There is nothing that this like is not really materialized. It's not something that exists. Well, I'm not stressing here how many knowledge graphs we have. You probably all are dealing with this, and they're getting bigger and bigger and more complex. These are very generic knowledge graphs. They're like Iago, Freebase, DBpedia. They're publicly available, and we can download it. That's why in research they're actually quite well used. So my personal vision, and this is what I'm pushing a lot, is actually, and I'm working with the companies and academia, like, so I'm doing fundamental research, but also focusing on the application scenarios, is that we should have a whole stack of technologies, starting from interactive algorithms, so algorithms that really suggest you something and you can get trust on the algorithm. You should also have some similarities among the entities that are explainable. And what we touched upon before, also in the previous talk, is adaptivity, right? So if I have a structure below like a Neo4j database, how can it change over time? Because changes will happen. Plus, it should also be personalized. They should be changed according to the user interest in some part of the data. And this is what I will talk about today. So I will talk about the exactly personalized part. So we realized something very simple. Okay, the user typically asks questions, right? As I started from the beginning, is Norway in EU? Then you basically can imagine that you are highlighting some part of your graph. Where is Venice located? Well, Venice is located, for instance, in Italy, but you can also imagine that starting from this specific node, you can be actually interested in capital of, in member of, and so on, so forth. So other connections. But the fundamental question is, can we find a summary, which is a subset of all the possible connections, all the possible entities and relationships, such that we can serve queries, not only now, but also in the future? So can we model somehow what is the user preference? That was a very interesting question that we asked ourselves. And we started by asking, okay, what is even a summary? So how can I define a summary? I don't want to go into details, but you can imagine that you have your own knowledge graph, any knowledge graph. You have a set of cipher-like queries, so a set of queries asking for specific relationships, arbitrarily complex, we don't know. You have a search cipher engine, so you have Neo4j serving the query and returning you the answers. And at some point, you put a constraint. You say, okay, well, if I'm a doctor and I have and I don't have any connection to the server, or the server is extremely slow, can I have a summary of that in my mobile? And so the limit is k, which is the number of edges, the number of triples, so entity, relationship entity. So it can be formalized and say, okay, I would like the summary that approximate my queries the best way possible, and the summary that in the future will answer also other queries. This is not an easy question. So how do I know about the future? So somehow it becomes as I learn from something that I've seen before. So and this is basically if you want, in a more complicated manner, how do I define this function? What do I say it is actually a good summary? And this is the problem, is that I don't know this probability. I don't know whether a node will serve a query or not. Is that specific entities should be part of the summary or not? That is the main question now. And what we did is, actually, you can use something that is also in Neo4j, so personalized page rank. Personalized page rank works, okay, a bit of more details that are not really important. It works in the following, right? You start from a node, from any node in any graph, and with the equal probability you choose the next node, and you keep repeating this process a certain number of times. But recording also the fact that with some probability you jump back. So you can imagine that this is wandering around and then coming back and coming back. In the end, you will count how many times you hit a certain nodes, and this will give you an indication of how important a certain node is according to the structure of your graph. Of course, there is a very beautiful mathematical expression, but basically it can be done in any graph analysis tool. This is one of the basic stuff. And therefore, we can estimate the probability that a certain entity V belongs or can serve the queries, can actually answer the queries. And this is given by two factors. So the historical, what we have seen before, what queries, what the searches have we seen before, and the fact that we exploit the structure. And this is something that sometimes we forget about. That below, when we construct our knowledge graph, we have a lot of different connections. So why not exploiting these connections? And so you sum all together. This is just a parameter that you set, basically. But the idea is that you can freely walk over the graph. And by freely walk in a very free manner, really randomic manner, you end up having something relevant. Well, what we have done is we lacked the real query logs, so we didn't have sequence of queries from users, so we generated, but we generated in a way that they were simulating what a user could do. A user could actually stay in the same topic. So I'm searching for patients with diabetes, and I stay with her. Or I keep changing. I search with this patient with diabetes, patients that that have cancer, and so on and so forth. So I keep changing. And the more you vary these two parameters, the harder it becomes the problem. This is also motivated by the fact that we have seen in other query logs, not in knowledge graphs, that was happening actually. And so we have basically extracted this sort of information. So we ask ourselves, is this working? Well, it turns out that it does. And it does by only, so if I take 10% of the graph, of our knowledge graph, we can predict all the queries that we had. So nearly perfectly. So this is basically 100%. And if you take 1%, so it's 2 orders of magnitude. If we take 1%, we have 80% of the times is correct. So it means that out of 5 answers, 4 are correct. And it's also quite efficient. So it's actually very efficient. Okay, I will not stress. So here is the experiments that we did with few topics and with many topics. So whether you're interested in patients with the same disease, more or less, or the same groups of proteins, or you keep changing over time. And you see that this is a harder scenario, of course, and the performance goes a bit lower. So the beautiful thing of this method is it is very simple. It can be easily implemented. It can actually these summaries are virtual graphs. So once you can load them, and then you query the virtual graph instead of querying the big graph, and can be used with any type of queries. We didn't port it in Neo4j, but it is possible to do it. So all the methods that we have used are already there. You just need to put them together. And we have the code that is available and can be actually used by anyone. I'm not sure I will be able to talk about also the second problems, but we are also working on something more specific, which is entity summarization. So if I talk about one, if I'm interested in a protein, or if I'm interested in a specific patient, or in this case, I'm interested in Denmark. The amount of information that we have is enormous, even for a single entity. We need a summary of the single entity. So if you see, for instance, in Denmark, you have a huge table. So you just look in Wikipedia, and all of these are basically attributes or connection to other entities. For instance, it is in Europe, it is in the European Union. This is a problem known as entity summarization, and it will be actually very useful for many different sectors. We are working currently with doctors, and this is a practical scenario that we're having, but also biologists could also be interested, and journalists alike. For instance, to find quick information about the news, or a small account of protein informations. So I don't have specific results, but we are getting some preliminary results now that are encouraging, and we are devising a more powerful method, and also that works if you haven't seen the entity at all. Okay, so I have only a couple of minutes. I want to just swipe very quickly about another problem that we solved, but I mentioned similarity search. Similarity search is, are there other cities like Copenhagen? Typically nowadays, you fix an algorithm, assuming that we know what these algorithms are, and these algorithms return a score for each of the entities related to Copenhagen. And for instance, it can rank the cities and says, okay, I return Bristol, Aarhus, and Berlin. There is another method, but this is, of course, doesn't involve the user. So the user doesn't know why the method returned these answers. There's another method which is called active search. So the algorithm selects an extra item and asks the user, is this relevant? So do you think that this is relevant? And slowly it learns, very slowly. So what we have done, and what we have asked ourselves is, can we do better? So if we have the static version where everything is returned at once, and then we have a very dynamic version, but it is returned in a very, very slow manner. Imagine you as a scientist looking for some specific protein, and then it takes hundreds of questions to be answered. This is not possible. So what we have done in a previous work, in a very recent one, is to combine these two. Okay, so if the user tells me it is interested in Copenhagen, well, I know that there are other cities around, so I use the similarity that I already know, and I use it as a second feedback somehow. I have the user telling me yes or no, and I also have a second opinion, which is of lower quality, which is a similarity that I already know, but that allows me to understand a bit better. Instead of looking around everywhere, I look to what this is suggesting, what this similarity is suggesting. And this allows me to remove a lot of candidates. Technically speaking, it's basically blending two different information, source of information. And this is done in an elegant manner with some probabilistic approach, basically. What we could see, and here I'm basically concluding, what we could see is that it was possible to really dramatically reduce the number of interactions. You see here that this is the budget, which is the number of questions, and the methods required to go up to 100 questions, but we were going down to 20. So it was quite an interesting improvement. Again, code is available. This is a bit harder though to be implemented in Neo4j, or at least I don't have the entire idea, but it can be, for instance, implemented in Java and then use Neo4j as a backend. So as I said, I'm working with other very interesting people, academia and industry alike. We are aiming to improve how people search in these knowledge graphs, how people explore data in these knowledge graphs. And we are going further and further with this vision, and hopefully in a few years we will have a fully working prototype. For now, there are pieces of code that have to be put together. Okay, this concludes my talk, and I'm very happy now to take any question from you.
Fantastic, Davide. Thank you so much. Thank you so much for your presentation. Really interesting approaches, and I think especially the last one, collecting user input for searches, or kind of aggregating user input for searches and all that. I think this is very, very relevant for a lot of Neo4j projects, right? Because the more users you have, the more, I don't know, brains you can essentially tap into and get expert knowledge out of that. Mm-hmm. Since this whole workshop is about collaboration and networking and so on, you mentioned that next to the basic research, you have active collaborations already. Do you have anything in the life science or healthcare space?
Yes. So one of the projects is exactly with hospitals here in Denmark, and they're interested in having. So they have we are constructing actually a knowledge graph with patient records and disease and what we know. Currently, we have a bit of problems with privacy here, so there are a lot of privacy issues. But we are trying to do our best with what we know and what they could provide as an information. And the purpose there is to find, so to help the doctors that are typically going around and clicking around in this, finding journals and hospitalizations, finding opinions on disease and so on and so forth, to put all of that together in a very compact manner so that they could actually go around with their mobile phones and check about informations. And another work, it's actually on biological data or gene therapy, and there they are trying to reduce the number of experiments in order to construct a certain the cure of a disease. So and they also have a knowledge graph which is very similar to many that that are also in this workshop. So connecting proteins, gene, and so on and so forth, and other informations about these specific experiments. So actually, yeah, so the applicability of this is very broad. So it can go from journalism to life science in general, and I'm very happy actually to collaborate. So slowly, I'm also building up some knowledge about about biology that I didn't have before.
So you have active collaborations in this area already. And,
yeah, but I'm very happy, of course, to take. So they're kind of limited also because one the hospital project I was put inside as a collaboration later on, but it is really broad. The question is really big, and so yes.
Yesterday we had a talk from Northwestern University about mapping OMOP data from the OMOP common data model to Neo4j. So maybe that's also interesting here. So there was one question about your methods. For such a big knowledge graph, I think you have to pre-calculate all the similarity coefficients. Am I right? And if that's right, how do you update it when new entities come in?
Yeah, that's a very big that's a very nice question. We currently don't update the similarities. You can approximate them. So especially if you're using PageRank, you can use easy approximations or sampling. That allows you to some degree of updating, but for now we assume that it is static. So it is quite challenging to update the weights somehow to what you know. We are working in the direction of update everything, so it's to make it more adaptive, but it requires quite a lot of not engineering, but it's a lot of sampling methods. So you have to imagine that you have to maintain a distribution more or less, and that is, if a new node comes, it changes where in the distribution is changing. So there is quite a lot of work to do.
So also from the algorithmic and analytical perspective, the whole update question is very relevant and still an open issue. I would have a more generic workflow question. So you use a lot of tools and graph and network analysis tools and frameworks and so on outside of Neo4j. So what do you think? How does the interaction of like these existing data science tools with Neo4j work, and where would be room for improvement here?
Well, there are there are some tools that are written in Java, and then you can basically already interface with Neo4j, and that we have used in the past. We have also, so most of the times in which we need the heavy computations, we really need to do that in Java. So we basically load the portion of the graph that we're interested in memory, and then we make the analysis, because otherwise making a lot of queries in Neo4j will be too slow from the computational point of view. And but basically you use a two-step approach. So you load some part of the data, you make your analysis, and then you save on Neo4j again. So that will be the workflow that we do.
Okay, because then we had that question a few times yesterday, because the GDS library can do a lot, but obviously we've seen so many examples of other data analysis approaches, and that question came up a couple of times. Okay, so thank you so much for your presentation, for your talk. Thank you so much for being here. I hope that you will find new ideas for collaborations, new projects, and so on. Yeah.
Thank you, Martin. And of course, if anyone wants to reach me out, is my email. You can just type my name and you will find my email. So, okay.
Perfect.
Thank you very much.
So much. And with that, we can hand over to our next speaker.
Hello.
Fantastic. Seshan Gori from IQVIA will talk about a graph database that he uses to evaluate drug safety data. So looking forward to your talk. Back to my healthcare, more patient-oriented studies again. So stage is yours. Looking forward to your talk.
Great, thanks a lot. So let me just see if I can share my screen. There we go. All right, hopefully everyone can see my screen. So today, this talk is going to be about how we can use Neo4j to help us evaluate drug safety. So let's jump into it. So to start with, the first question obviously is how do we evaluate drug safety? What do we mean by drug safety? So all the drugs that you and I use as patients, they've all been through extensive clinical trials. They've been approved by whoever your local regulator is, so the FDA, for example, in the US. However, even once drugs are approved and on the market, we still have to have some way of identifying if anything's happening that shouldn't be happening, what we call adverse events. And these adverse events could be very mild, something like nausea or fatigue, or it could be something much more serious like a stroke or a heart attack. So in most jurisdictions, there is some kind of spontaneous reporting system available for recording adverse events. So this is a system that allows physicians or patients to be able to record if they took a drug and they had some kind of unexpected side effect or adverse event taking place. And the best known of these is the FAERS system, which is operated by the FDA. I think that stands for the FDA Adverse Event Reporting System. And that's the system that we're going to be looking at. So if a drug has adverse event reports, should we be concerned? And the reality is every drug is going to have some entries in the FAERS database. So the real question is, how do we know if this represents some kind of potential problem? And the typical way we deal with this is to use odds ratios. So I'm not going to get into the detail of the statistics, but essentially what you look at is you build a 2x2 table. You want to know who's been taking a drug and had the adverse event, who took the drug and didn't have the adverse event. And then you look at all the patients that didn't take the drug. And again, you look at how many of those had the adverse event and how many of those didn't have the adverse event. And then you can look at a ratio of the two. That's one statistic. There are other statistics you can use as well, other ratios, to understand whether or not we think there's a link between taking a drug and an adverse event happening. But there is a slight problem with this, which is that the FAERS data and any other spontaneous reporting system data is going to have a certain amount of bias. And that bias is going to be the fact that the system's only going to contain data where adverse events have occurred. So normal drug use, where the drug gets used, nothing goes wrong, is never really going to turn up in this system, except if you happen to be taking a drug alongside another drug. So what we need to look at is what we typically call real-world evidence. And by real-world evidence, I mean here information about how the drug is being used in real life. So an example here could be looking at hospital electronic health records. We could look at insurance claims data as well. And indeed, that's specifically insurance claims data is what we looked at with our solution. So the first problem that you run into, and a few of the previous talks have touched on this, is that every single database you use is likely to have be using different vocabularies to record drugs and conditions. So, for example, in the FAERS database, all of the adverse events that occur, the reactions when you take a drug, are recorded using a vocabulary called MEDRA. In our system, where we've got insurance claims data, we've got everything sitting using a ontology called ICD-10. So we need a way of mapping between the two. And fortunately, the Odyssey initiative provides a way of mapping between many different vocabularies. So you can see here on the right-hand side, we have here all of the MEDRA hierarchy. We can go from there to SNOMED, and from SNOMED we can get down to, in our case, ICD-10 CM. So this makes life a lot easier. What's important to note, there is still a certain amount of messiness involved. It's not quite as clean as being able to map from one thing to another directly, and some of the mappings may be missing. So you can't rely on this 100%, but it's probably better than anything that's out there at the moment. So what did we do? So we essentially took FAIRS and the real world evidence that we had. We combined this with the Odyssey ontologies, and we did this all using Neo4j to connect them. And this gave us a solution which lets us do these odds ratio calculations both over the FAIRS data and then find the corresponding real world evidence, in this case insurance claims, to do the equivalent calculation there to see how the two compare. So let's talk a little bit about how we actually implemented this. So you see here a model that we developed and we populated with data in Neo4j. So just for example here, it's probably a bit too small to read, but in the center here we have a drug. In this case, it's metformin. It's a common anti-diabetic drug. This is encoded in RX norm, which is a drug ontology. From here in the dark blue, these are the entries in the FAIRS database for people who are taking that drug. And you'll notice here, this drug could have various different brand names. Then for anyone who's taking this drug, this connects to the case record in FAIRS. So each of these has got a different case number. From there, you can jump to the reaction that happened as a result of, or potentially as a result of taking that drug. We don't know whether there's a causal relationship here. All that's recorded is somebody took this drug and they had these reactions occur. So you can hear here, for example, pain is a very common one. We've got malaise here. So the different reactions that happened, and then from there we translate this into, in the green, we have the entry in MEDRA. And what I haven't included here to save space is then from MEDRA, we would go to SNOMED, from SNOMED, we would go to ICD-10, and that's what lets us eventually connect to our real world evidence. So, we loaded the ontologies into Neo4j. So we've got all the FAIRS data sitting in Neo4j. We've got all of the ontologies from the Odyssey project sitting in Neo4j, and then we linked the FAIRS data to those ontologies. Now we come to our real world evidence, our insurance claims. So the insurance claims data is very, very large. We're talking billions of rows of data. We did initially look at pulling this into Neo4j, and we figured out that really wasn't a practical solution. I mean, you'd need a ridiculously large amount of memory on your box to make this work. And actually what we realized is we're better off leaving this data sitting in place in the Hadoop cluster where it lives. So there's already a whole bunch of ETL processes that populate it, and we didn't want to have to replicate those either if we're pulling that into the graph. So yes, didn't make sense to move that into Neo4j. So what we did in practice is we used the graph to generate the ICD-10, those are the condition codes. We used to generate the corresponding NDC codes. So NDC is the ontology we use with our insurance data to record the drugs that patients were taking. And so we take the FAIRS data, we convert it into a series of codes in the ontologies we want, and then we feed that back into Spark to query the Hadoop cluster. And all of that's orchestrated using Python. So that seemed for us to be the most efficient way to get this to work. And to be honest, we put the FAIRS data into the graph database. You could equally do the same thing with the FAIRS data. You could have that sitting in a relational database and really use Neo4j to hold the ontologies, the graph-like data, which is what it's best at doing. And then once we had all of that in place, we built a React front end on top of this to give us a SaaS product, which we can use to run the calculations for any drug or adverse event combination. So let's have a look at some of the sample output from what we built. So as you can see at the top here, let me just switch to laser pointer. Okay, so you can see at the top here, we can pick a drug, we can pick a date range. So this drug here, we're using the RX norm ontology. So you have to pick something from the RX norm ontology, but you could equally use any other ontology if that made more sense. Once you've selected that particular drug, we're able to then bring back from the FAIRS data all of the adverse events. And in this case, what we've done is we've grouped the adverse events using one particular level of the MEDRA hierarchy, which is HLT, high level terms. But you could equally go higher up. You could go to SOC, which is the system organ class. And that's part of the beauty of having all of this connected with a graph. It's very easy for us to aggregate at any level of the ontology that we want. So this has brought everything back at the level of the high level terms. We've picked here, so in this case, we picked aspirin, we've picked renal failure and impairment as the high level term. We can then, based on selecting that, we can bring out all of the individual preferred terms. So this is the level that's recorded in FAIRS. And we've selected here the top one, which is acute kidney injury. So what we want to know is what's the relationship, or what kind of odds ratios do we get for patients who are taking aspirin and had acute kidney injury. So once we've done that, down here at the bottom, what we can do is we can do the odds ratio calculations both for FAIRS. So this data is all sitting in Neo4j, and we can then jump over to the data sitting in the Hadoop cluster based on the codes that we're sending over to get the same calculation for all of the data in the insurance claims. And then on the right-hand side here, you can see the comparison between the two. We've looked at two different ratios here. There's the PRR and the ROR. They're both different odds ratios that are used when analyzing drug safety. Now, what you can see here, and this is quite a common thing that will happen, is that the odds ratio comes out much higher in the FAIRS database compared to when you look at what's happening in the real world data. And this is not surprising. The FAIRS database is obviously, as I mentioned earlier, biased towards people who are submitting details where they had some kind of suspected adverse event, whereas the data we have sitting in our real world database is showing much more typical usage of the drug, where it looks like there doesn't appear to be anywhere near a strong link between taking this drug and this particular adverse event. And if you go to PubMed, for example, you'll find sort of dozens of papers on people who do this kind of analysis over FAIRS data. And what a system like this lets us do is then be able to do that same comparison across real-world data sets. As I mentioned, we're looking at insurance claims data. You could equally look at, we haven't done this yet, but we can connect it to, for example, electronic health record data or any other data set that we wanted to, as long as there's a means to be able to link from one ontology to another. Now, having everything sitting in the graph also allows us to do a couple of other quite interesting things. One thing that we can do is we can, for a particular drug, we can compare that drug's safety profile to other drugs in the same class. And you may have a question about, what do we mean by class? Again, this is coming back to the ontologies. We can pick an ontology. I mean, ATC, for example, would be a useful one to use here. So we can then jump across from whichever ontology we're using for the drug, NDC, for example. We can go across to what the entry for that drug is in the ATC ontology. We can go up one level to find the drug class. We can come back down to find all of the other children, so the siblings of this drug. And then we can do the exact same calculation that we've done here for every other drug in the class. And we could do it for the class as a whole. And the reason you might want to do that is a drug may well have a certain risk of an adverse event, but maybe that risk is no worse than other drugs in the same class. That's probably the more useful thing to look at. And then the other thing that we can do, because we've got all of this linked sitting in the graph, is that we're able to look at combinations of drugs. So if we have reports where multiple drugs are being used, we can do that same analysis looking at that in FAIRS. We can look at the same thing again in our real world data. So those are some of the sort of the more interesting areas that we can move on to after this. But the main advantage here the graph is bringing is the ability to be able to connect multiple heterogeneous data sets and then be able to do a single analysis across all of them. And so yeah, that's about all I had to cover. I'd love to hear if anyone has any questions or comments on any of what I've said.
Fantastic. Thank you so much for your presentation and sharing your application and knowledge graph and analyses. So let me jump over to the chat and see if we have questions. And while I do that, I would like to again reuse the question we have after every talk. How do you work with data updates? So we had the question a couple of times when it was about genes and proteins and so on. And I think in a certain way, the terminologies and vocabularies are very fundamental to everything in medical informatics, right? But they change. And how do you handle that? How do you deal with that?
Yep, no, that's a great question, and our team was chatting about this morning actually because we'd seen the same discussion on the other talks, and the reality is the easiest solution for us is just to wipe the database and to rebuild it. Is much easier than trying to come up with some kind of incremental process to fix it as we go along. Is the simple answer. I suspect as the field matures, there may be better solutions in place to have incremental updates, but for now, we don't have anything more sophisticated than that.
Mm-hmm. So you pull the latest version from the Odyssey vocabularies.
Yes, exactly. And bear in mind, a lot of this data doesn't change that frequently. But yeah, if we need to, we'll just reload it all again.
Mm-hmm. And maybe a follow-up question about the vocabularies. So as far as I know, the MADRA-SNOMED relationships in the Odyssey vocabularies are not complete yet. So I think MADRA is only used for classification, but correct me if I'm wrong here. Did you use other mapping data sources on top of the Odyssey vocabularies?
So we didn't use additional data sources, although that is obviously an option. And so typically when this kind of analysis is done, if you go to PubMed and look at any of these papers, typically what will happen is someone will just manually go through and do the mapping between one coding system and another. And what we're trying to do is come up with a more general solution. So I mean, there's a decent level of coverage of MedRA preferred terms at the PT level. But it's not complete. I mean, so what we did is go to SNOMED, jump up a level, and then go back down again to try and get reasonable coverage of the relevant codes. But yeah, so I think with the Odyssey model, that's probably about as good as you can do with the data that's there now. Or the alternative is to find other mappings.
Mm-hmm. But at least if it's in a graph, you have the option to extend it. Mm-hmm. Again, the same question we had after almost every talk. I mean, there was the OMOP mapping talk yesterday, and we had a bit of discussion after that talk. Do you think for this very fundamental space of terminologies and vocabularies, do you think there could be kind of a default way, a default graph representation of this that we could reuse?
Sorry, could there be a default representation of what?
So we had a talk about mapping OMOP data to Neo4j yesterday, and we had a bit of a discussion. So the power of OMOP is that it's a common data model, right? We all store data in the same way. Do you think there would be something like a 100% correct default way to represent data from OMOP vocabularies plus events in a knowledge graph?
It's a good question. You're right, as in it feels like there's a lot of us doing the same kind of implementation again and again and again. I suspect there are probably certain decisions that are made in modeling which are probably fairly application specific. Is the reality right now? That said, it would be great if there was a default off the shelf. Okay, click a button and you get all of this pulled into a graph, and I guess maybe that's something that might make sense for Neo4j or someone else to do. I wouldn't be surprised if people have to tweak it for application specific reasons. Mm-hmm. So I mean, one simple one actually is that we were looking at. This is probably not critical, but for example, with the OMOP model, you'll have all of the ancestors linked at every level for each ontology, which is done primarily, I'm assuming, because if you're loading this into a SQL database and you want to find, you know, the parents, the grandparents, the great grandparents, you can do it with a single join. Obviously, if you've got it sitting in a graph database, you can just traverse the links to do that. So it's not really necessary. So there are some possibly simplifying things that could be done if you're loading this into a graph.
Mm-hmm. So there needs to be more work and more discussion in the audits community, essentially, how this should work and how we can standardize it. Okay, there's another really interesting question from LinkedIn. A great presentation. Are you using this approach also for multilingual insurance data? And if yes, how did you do the mappings across taxonomies?
It's a good question. So we're not doing everything we're looking at so far. We've been looking at the US only, so it's all been in English. I mean, in theory, if you're using one of these ontologies or these coding systems, I would have thought it would be fairly language independent, but I don't know for sure. We haven't looked into that yet.
Mm-hmm. I think you will be surprised about the differences and so on. But I mean, this is what the Odyssey community is all about, right? Mm-hmm. Also including vocabularies from different languages. Okay, let me jump back to the chat and see if we have other questions. There is a question about the vocabularies. Do you also use, or what about the use of UMLS?
So we haven't been using UMLS. I mean, we essentially just took the Odyssey mappings off the shelf and just took the simplest parts we could find between the vocabularies we needed, and I guess as we find holes, then we might look to use other solutions as you mentioned.
Mm-hmm. Okay, let me quickly scroll to our chat and the Q&A section. But I think that was the last question. So a lot of questions about the vocabularies, also multilingual and so on. So Sishan, thank you so much for your presentation. It was really interesting to see more approaches, more projects from the healthcare space, or let's say from the patient level data. Thank you so much. I hope you enjoyed the workshop and the other talks, and I hope that you can also find interesting ideas, input, and maybe potential ways to collaborate. So thank you so much.
Thanks.
And with that, I would like to hand over to our next speaker. In our next talk, Emre Varol from John Snow Labs will present I think, very interesting topic that came up a couple of times yesterday. Will present how he builds a clinical knowledge graph with Spark NLP and implemented, obviously, in Neo4j. So, Emre, great to have you here. Really looking forward to your presentation. The stage is yours.
Thank you, Martin. Let me start and share my screen and test it. Am I sharing?
Yes, we can see the screen.
Okay. Thank you very much for a great intro. I really appreciate it. Hello, everyone. I'm Emre. I'm based in Istanbul, Turkey at the moment. I'm a data scientist in Johnson Labs, and here is the topic: creating clinical knowledge graph by Spark NLP and Neo4j. And here is the agenda, and I'll start explaining Spark NLP at first, and then we will continue with Spark NLP in healthcare, how it works, and the pillars of it. Next step will be our other main component to create knowledge graph, as you know, Neo4j. I think most of the attendees have some background info about Neo4j. After quick remind, I will jump into clinical knowledge graph. What should be the properties and features of a clinical knowledge graph and the points to be considered by the practitioners. And then the last one is here is the most important part of this talk, how we can create clinical knowledge graph by Spark NLP and Neo4j. We will do this part live. It will be live demo part. And let's start. So Spark NLP is an open source NLP library released in 2017, and right now it has around 40K daily downloads from PyPy. Our monthly download is about 1.2 million, and totally we hit the 10 million already. We support 4 languages: Python, R, Java, and Scala. The goal was to be able to create a single unified NLP library that will require no other dependencies other than Spark itself. And it should also work on the clusters. By the way, as far as I know, there was no other library that could support to run on Spark clusters, NLP libraries. We also wanted to take advantage of the transfer learning and implementing the latest and greatest state-of-the-art algorithms. As you see, we have public and healthcare models. Healthcare models are licensed, which is not free, but the public is totally free. We also have another library, which is called Spark OCR. As you know, mostly data entry starts from like PDF or images when it comes to clinical text. So EHR, electronic health records or medical records are usually go through Spark OCR for that, but I'm not going to cover that today. On the left-hand side, on this part, you see the public modules. We are supporting more than 200 languages by using the same NER architecture, which is another nice point to underline, by the way, because we can switch one language to another easily as long as we have the relevant word embeddings or the word coverage to accomplish certain tasks in NLP. We have some deep learning-based emotion detection or text classification algorithms or pre-trained models that you can just plug and play. So instead of trying to build your own pipeline, you can just download and use our pre-trained pipeline and then just feed your data frame. So you end up with the latest state-of-the-art resource using which were various word embeddings, including GLOW and the latest cool kids in the NLP town, BERT, Elmo, Albert, Distilbert, so on and so forth. So we all support them, and you can just use any model like a building block and like a plug and play. On the right hand side, you see clinical versions. Like public models, we have 4 main clinical models. We have more than that, but let's just cover 4 of them: clinical entity recognition, clinical entity linking, assertion status, and relation extraction. We have around more than 50 different NER models. Clinical NER models, which means like pre-trained on clinical NLP data sets. that are being used in clinical NLP challenges like I2B2 or N2C2 datasets, we call them. And I mean, we call these models like anatomy NER, pathology NER, I mean, PHI NER, etc. To extract the meaningful chunk given the task, clinical entity linking is assigning some SNOMED, RX norm, ICD-10 codes to these entities detected through NER. So the assertion is very important model in healthcare domain. It's like if the clinical node is talking about the patient's father, not the patient itself. We need to know that information so that we can assign different features. I mean, maybe we will create different features accordingly, or if it is not that, we can just filter out those symptoms maybe. For the de-identification, here, this part, we remove or mask the sensitive information according to HIPAA rules. We need to hide or conceal some of the information, and these are called, sensitive information, and we need to de-identify or obfuscate. Obfuscate means like replacing the fake names or entities for the real ones, okay? So we also have some pre-trained models that you can plug and play and then combine with the public ones. I mean, you can use these models for word embeddings, and you can combine with the clinical models. We keep Spark NLP up to date and upgrade in every 2 weeks with releases. We have released more than 75 times. The main focus is here to have a single unified library for all NLP and NLU needs. And also we have Slack channels, John Snow Lab Slack channel and GitHub, on the right side, you see some NLP features and comparison of the other libraries. I mean, here is the Hugging Face, Core NLP, NLTK, Spacey, and our product Spark NLP. Let me show you an example to answer the question, what is the output of the Spark NLP models? I mean, the text here, clinical text here, a 28-year-old female with a history of type 2 diabetes mellitus, diagnosed 8 years ago, takes 500 milligram metformin 3 times per day. As you see, the first NER is age of the patient, and the second one is gender. And as you see, the clinical finding, type 2 diabetes mellitus, and we can use our assertion model and also our resolution models to map these clinical finding to SNOMED code, ICD-10 code, and UMLS code. And after that, you see some date, relative date here, 8 years ago, and we can extract these using our NER pipelines. And the dosage of the drug, the metformin 500 milligram, and we can map just map these drug to our the RX norm codes and the NDC codes, and you see the frequency of the medication. Also, we can calculate the HCC risk adjustment score according to these kind of clinical text. And let's talk a little bit about the NER in healthcare side. As you know, everything starts with NER because it is the minimum meaningful chunk of any clinical text. The importance of NER might not be that much in any other domain, but in healthcare, that's very important. Everything builds upon clinical NER models, name entity recognition models. We have many other models that have nothing to do with NER, but when it comes to extract some knowledge to get a sense of what is going on inside the document, we start with clinical NER model and then use the other models. use the output of NERs as an input, so that it could assign something instead of dealing with the entire text. Entity linking, assertion status, the identification, and relation extraction use the output of the NER models. For an example, assertion status model assigns some status if the fever and sore throat is a problem entity coming from NER. Association starts to find it is absent or present, given the text, given the sentence that Chang lives in. One more, relation extraction models try to find relation between NER chunks. So that why NER is very important. I explain why NER is very important and highly valuable in clinical texts. Here is another example. In my humble opinion, annotating the data and NER is the brain and the heart of the NLP body that manages every other tasks and subtasks. Recognition of the name entities is basically classification of tokens. NER tries to locate and classify predefined categories such as persons, locations, organizations, hospitals, medical centers, medical codes, measurements, units, monetary values, percentages, quantities, and etc. We use NER to downstream the related tasks to answer real-world questions. Which hospital and department have been admitted by the patient? Which clinical tests have been applied to the patient? What are the test results? Which medication or procedure has been started? And here is the example of relation extraction. As you know, relation extraction is the task of predicting semantic relationship from the text. Relationships usually occur between NERs and NER chunks, and the RE, relation extraction, is the core component to build relational knowledge graph. It is essential for NLP applications like question answering and summarization, etc. Clinical RE plays a key role in clinical NLP tasks to extract information from healthcare reports. You can use it for multiple purposes, such as detecting temporal relationships between clinical events or drug-to-drug interactions, the relation between medical problems to treatment or medication interactions, etc. I will not go deeper into the importance of the RE in medical studies today, but I think you know about it. In the healthcare world, the automatic extraction of a patient's natural history from clinical text or EHRs is a critical step. It helps to build intelligent systems that can reason about clinical variables and support decision-making. Any intelligent system should be able to extract medical concepts, data expressions, temporal relations, and the temporal ordering of medical events from clinical text. But this task is not easy. I mean, it is very hard to tackle due to the domain-specific nature of clinical data, such as writing quality and the lack of structure, and also the presence of the redundant information more generally. I think we can handle this task by combining some rule-based methods and the unified NLP library and the power of knowledge graph. In my opinion, this combination is the most appropriate and the quickest way. I mean, here is the brief introduction to Neo4j. Neo4j is a highly scalable native graph database. The core belief behind it: connections between data are as important as the data itself. I'm pretty sure that using the connections between data will create a competitive advantage to produce actionable insights in healthcare. And you see some, some information about what's graph database, and I will skip this part quickly to save the time for the live demo part. And you see some other informations about the Neo4j and Neo4j graph. Cypher is the Neo4j's graph query language. It's declarative pattern matching language. I mean, this part is important, pattern, because it's all about patterns. It has SQL-like syntax, and it is designed for graphs. This is a basic pattern, what if we only want the relationships of a specific type. As you see, A acted in M, and the actor acted in the movie. This is like two nodes and one relationship, and we try to get these pattern from the clinical text to achieve our goals. And you see the Cypher query example about this this graph, okay? And when we start to talk about knowledge graphs, and here is an important distinction. These are charts, not graphs. So it's good to start using the right terminology, and we will jump into what is clinical knowledge graph. Healthcare data are complex, as you know, exist in multiple places, and the redundancy, structured and unstructured, inconsistent definitions. You can find inconsistent definitions in the clinical text, and also the regulations are changing, and we need new requirements. So for the clinical knowledge graph side, we need to create data points which accurately represents the patient history, rather than creating a patient history which contains a variety of data points. And it should be a representation, structure, and method, and it should organize the entire body of clinical knowledge. It should contain high relevancy in clinical data. It should be universally accessible. And these are the properties and the features of the knowledge graph. This illustration is one of my conceptual graph models. Here is the tip of this talk, especially for clinical ones. Before creating knowledge graph, you should spend some time working on conceptual graph models. Otherwise, it is very easy to create graphs that are irrelevant to your purposes. It will be completely mess. I mean, you have a big chance to connect irrelevant entities to each other. After creating the knowledge graph, you should validate the graph by querying and cross-checking with the raw data, like the input-output analysis. And here is the one of our knowledge graph for the text on the right side. You see that text. I mean, we extract this. I mean, we create this knowledge graph from this clinical text. And as I said before, I spent some time to create this. The conceptual graph model. To create this model, because this is not very easy. As you see, we have level 1, level 2, and level 3, level 4, and level 5. And also this one, level 7. Okay, I mean, you have to always keep in mind that before creating knowledge graph, what should be our the conceptual graph, and how can we map the clinical text into our conceptual graph to create the knowledge graph. I'm done with the presentation side. I will just reshare my screen after stopping it to jump into live demo. Just give me a minute, please. Okay, I think I'm sharing the screen. So we can start with the live demo part, and the you can find these are I mean these you can find this notebook in our workshop repo in the GitHub. If you want, I can share the links with you, or you can just jump to our workshop here. Can I send message from the to all people, all hands here, maybe? No, I couldn't. So you can find our repo from the GitHub, and the you can find the knowledge graph side here. It's it's 10.2. Okay, this notebook. You can find the details here, and I will just cover one example for the public side, one example for the licensed version. First of all, the licensed version. After you get this notebook, you can also get, I mean, the trial license from the Spark NLP. I mean, from our website, Johnson Labs website. You will get, you can, after filling the form, you will get 30 days free trial, and you have to load your license key before starting, because you will download according to these secrets. Let me just download it, and we will we will install all these libraries. It is very easy. I'm not just skipping this part, because I mean we also experiencing this part, I think, and you will see. I'm using Spark NLP public version 3.3.2 and the licensed version 3.3.2. We are using still the PySpark version 3.1.2. Okay, and here is the pipeline. First of all, we we have document assembler annotator, and after that sentence detector and tokenizer, word embeddings model, and the post tagger, NER model, and also NER converter internal. I mean, this is NER converter to make the NERs to chunks. I mean, combine them in chunks, and the dependency parser model and the relation extraction model. You all you can just create these are inside the code snippet, and you will just write the pipeline. I mean, you just put them in line like Spark ML modules. Okay, and we will fit the we will fit the pipeline with empty data, and after that we will use our text to feed it to get the results. And this is a get relations df, the helper function to get the relations of the light pipeline. And so, I mean, maybe you have a question, what is light pipeline? Light pipeline is the, I mean, you can get the results very quickly. I think it's 10 times faster than the normal pipeline. Here is the text, a 20-year-old female, I mean, blah, 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 you can read it. And I will just create the light pipeline using this pipeline. And I will get the relations with this helper function and show you as a pandas data frame. So when we run this, our pipeline, the pipeline has founded this one. I mean, let's check what is the pipeline. This is Phylogeny relation extraction pipeline. Phylogeny, I mean, we are working on the drugs, right? So we will see some relation between drugs and the modifiers of the drugs, like the duration and the route, dosage, so on and so forth. As you see, the first one is 5 day. The first chunk is 5 day and amoxicillin. The other name of the drug and for 6 months, and insulin, Advil, and the 40 units insulin, metformin, and the strength and the frequency of this drug. Okay. And after that, we need to connect with the Neo4j, and I will show you how you can open up a Neo4j sandbox. I mean, I will use a sandbox in the Neo4j site, and you can just launch a free sandbox from here. And after you launch it, you will see a window. You will just choose new project, and after that, you will see a blank sandbox here, which is not available for now because I'm using it in here. And you will see some username, passwords, and bolt URL here. After running this part, this code snippets, we will use a class for the Neo4j connection, and we need a helper function to create nodes and relationships in batches. And we have a function here, but I will skip it for now, and I will return back here. So this is the URI, password, and user. You will just copy and paste from the right side to left side, and you will create a connection here. Okay. So we can talk about what is the NERs and the relations. What will be our conceptual model, graph model, to put all these information properly in our knowledge graph. Okay. First of all, you will see the chunk 1 and chunk 2. These are related entities, right? And these are the entity types here. Duration, 5 day is a duration. Amoxicillin is a drug. And you will see the relation also here. Okay. But I will not use this part. You can also relate all these entities using dynamic relation using APOC. APOC, you know, the query languages for Neo4j, another query language. And what I'm doing here, first of all, I'm moving line by line, and when I get the chunk 1, I create an NER node here, and for the chunk 2 in another NER node, and I relate them using links keyword. Okay, as you see. And I also use the row relation here. Here is the row relation. I just check out what's happening in the what is the type of the relation between these two guys, these two chunks. Okay. This is I think the creating this kind of functions, to map the result of the NLP pipeline to our knowledge graph is the key point. And I will run also this function. And after that, first of all, I will drop all constraints, drop all nodes inside this sandbox, and I will create constraints on the NER nodes. And after that, I will run the my main function to put all the entities into our into our nodes graph. Okay, I'm sorry. So we are done. All our NERs and relations are stored in our in our knowledge graph here, graph database. Okay, you can just check out here. Okay, address, and you see the relations and the entities as you see. Okay, for this patient, she used metformin and the 2 times a day and 1000 milligram. Okay, like this. And also you can run some queries on this knowledge graph. As you see, you can extract the information, useful information from this knowledge graph. Maybe you can use these results as a Q&A. Okay, Q&A application in NLP. You know, it's very hot topic now. And we will just filter out this NERs, I mean entities, just on the Advil. You can get this one. So we can say that the patient used Advil sometimes, I mean the 12 units, 40 units, for 5 days, and 1 unit. Okay. And I will skip this part quickly because we are running out of time, I think. And this part is another part for the creating knowledge graph, but this is public one. Let me just run this part and get the. I mean, we can walk through the all components of this pipeline. Again, we have document assembler, sentencer, tokenizer, embeddings, NER tagger, I mean, and the NER chunker, post tagger, dependency parser, and here is the different component from the first one, type dependency parser. Okay, and the graph extraction. We will use graph extraction here. This is, as I said before, this is public one. So we will not use any licensed version of the Spark NLP. Okay, and we have again the some helper functions like the get graph results. I will create the graph and I will feed this text to my knowledge graph. Okay, you can just check out the text here. Okay, some guys, where they born and where they live for now, and where they work also, and there's some other kind of information about the countries, related countries. Okay, and we get the results, and we also used our helper function to put all the results into the pandas data frame. Okay, while it's running, let me show you the results, because I think it will take a little bit time, because I didn't use light pipeline here to show you the all the results. I mean, how it is working, our usual pipeline, base pipeline. When we run all these code snippets here, code cells here, we will get this kind of information. Jon Snow, born in England. Jon Snow lives in New York. Peter lives in New York, so on and so forth. Okay, and we will again run these helper functions to put all this information, graph information, to our knowledge graph. And as you see, the same works. I mean, I didn't change anything else in this function because this function can work with this structure. It can work for any kind of relation. I mean, output of the relation extraction models. As you see, again, same kind of things, and we will replicate the same, reproduce the same steps again and again. Okay. Oh, it's done. Let me run it again. You see the results here. Okay. I think there is something else in the Colab side because I couldn't see the outputs if I didn't click on the output side. And you will see the results here. And also we will check from the other part. These are all running, as you know. These are all running, and this is also the same. This is also the same. I will just run this part. I'm sorry. Oops. I will drop all nodes, create constraint on the NERs, and I will rerun this part to put everything on the knowledge graph side here. As you know, we are using the same sandbox to. I mean, we will just erase all of them. I mean, remove all the relationships and the NERs and just recreate this knowledge graph. Okay, you see the result here. Okay, I'm done. I hope everything is clear for you, clear for everyone.
Emre, fantastic. Thank you so much for the presentation. With a talk from David a bit earlier, we kind of had a good look in the broader, data analysis and data extraction and data exploration that comes after you get your data into the knowledge graph. And I think it showed us a couple of really interesting examples from the NLP area, how we can, enrich data, use data, and map it to a knowledge graph. Just one. And we don't have much time for questions, but just one quick question. So there is a free version of Spark NLP, right?
I'm sorry, I couldn't hear you very well. Let me adjust my speaker again.
Just a quick question. Is Spark NLP free?
We have public version and healthcare version. Healthcare version is licensed version. It's not free, but the public version is totally free, and you can use this graph extraction for the non-clinical text. Okay, I mean this is the example of the example for the public version. This one.
Yeah.
This part.
Okay, and then there's the healthcare version which is paid. Okay.
Yeah. When it's public version, free.
Fantastic. So thank you so much for your presentation. Thank you so much for the live demo. I think this topic is actually relevant for almost every other presentation that we have here because we all need data from text, right? And Evelyn nicely summarized that yesterday in her presentation. We need it because so much information is just hidden in PDFs, in PowerPoint presentations, and in text in general. So that was our last presentation for today and for the entire workshop, of course. And like we announced a bit earlier, what we would like to do in our closing session is to get a couple of speakers from yesterday and from today back on stage and discuss with them a bit how we can now take the next steps. Because we said at least 500,000 times that we want to do networking, collaboration, maybe share data models, share data, share our general analysis approaches and so on. But how do we do that now? So what are our next steps? And I would like to ask Alexander Jarash, Piyush Sahu, Rita Kolako, and Jeremy Kanya back on stage to discuss a couple of ideas how we can do that. And the reason why we asked you is that you represent very different organizations. So we now have someone from a pharmaceutical company, someone still fully engaged in academic research, and someone from the, Alex, how would you call it, data management perspective. So somehow sitting in between these two worlds. And if Piyush joins, we even have a biotech and chemical company. So Michael, you're of course also welcome to join us. Let's maybe start with a very, I think, very obvious question. So we all have genes and proteins in our knowledge graph, but we all have our own parsers, data loaders, and data pipelines. So what do you think? So this is a very practical, I think, idea or suggestion. Do you think it would be possible to agree on kind of a standard model for this super basic stuff, for this basic biology, and actually share something like our data loading pipeline? Just shoot. You're all live.
So I would answer the question first. I think sharing is caring, right? And I think we should really talk about sharing knowledge and also sharing the way of how we deal with the same entities, as Martin mentioned, like genes and transcripts and proteins, as well as ontologies. And I think the overlap is, let's say, 80 to 90%, and I think there are only some minor how do you say, some minor differences in the way of how we see genes, for example. I think it's a pretty straightforward way to work on a common data model for the molecular entities, for example. I guess that would be a good start, because we are all talking about the same genes, we are all talking about the same proteins. If we see that from cross species, that's the same thing, right? So we are talking about genes in bacteria and genes in viruses and genes in humans. And I think we could sit together and at least propose a graph model for these entities, so that we can reuse that and not reinvent the wheel at each and every company or academia.
Mm-hmm.
I completely agree.
Jeremy, sorry, just continue. Please continue.
No, I completely agree with Alexander here. I mean, of course, the basic idea is always the same. Central dogma is there. Of course, organism can be less complicated. So if you start with the human or even complicated organisms, you can always go low in terms of complexity, which will not harm overall project. And what I see in this workshop, that what is very important is, of course, bringing knowledge, right? So after making this a central dogma of gene, protein, transcript, and other things, I think there should be a standard way how we want to show relationships between these entities. And I think it was also mentioned already. So like how someone models a protein-protein interaction network, how someone models a regulatory network, and because we would like to in the end get something out of it, right? So a standard way of modeling would really help, actually, and actually give us a head start in terms of if somebody wants to build something.
I think that really comes down, like Alexander mentioned, to the data model. If we have one that can accommodate most of the general databases, the UniProt, the pathways, something like that, it can always be extracted as subgraphs, and people can build from it. So I think that really comes down to what we think is the basic common unit for most people and how we can design that.
Jeremy, you built and presented a huge knowledge graph with a lot of applications and a lot of queries and a lot of things that your users ask the graph. What do you think? Do you need the flexibility to model and do everything kind of in your own way, or do you think the core can be reused and shared?
Sorry, what's the question for me?
For Jeremy, but I think he's muted.
Yes, it's okay right now?
Yes, now we can hear.
Okay, cool. So there is a basic question, but it's not straightforward to model it. It's basically a gene, a transcript, or a protein, because basically the nation of gene, you can have information from UniProt, NCBI, ensemble, gene code, and so on that have their proper identifier system. So the way we model that into Pegasus is about, we have an entity gene coming from UniProt with cross-references from a gene that is the same coming from NCBI, ensemble, and so on. And we do the same thing for the gene, the transcripts, and the protein. So in our graph, we don't have a big entity gene that have plenty of meta information, but we split the notion of gene into a gene node, but that are coming from different databases. And one thing we are thinking about is about in a couple of data model you showed during this today presentation and yesterday is about a gene can give a protein through a process of translation and transcription. But it's not always true. It's depending on a cellular context, on a particular tissue, at a given step during the development, and so on. And moreover, I think in all of your graph, you have about 20,000 protein nodes, but there are plenty of protein isoform. So the notion of how can we model a complex object, such as isoform, is also an important question, I think. And yes, in plenty of databases such as Campbell, we have notion of a chemical can have an activity on a particular target. But when we are looking at the real scientific papers that describe the assay, it's not a monomeric form. It can be an aggregated form of the protein and so on. So really the basic notion of how can we model the genes, the transcript, and the proteins that are the basic notion of biology is not straightforward.
Mm-hmm. So with all this complexity that we need in these specific projects, Jeremy, what do you think about Alex's suggestion to not kind of reuse data loading or data parsing, but coming up with ideas or ways to share subgraphs so that you can, Alex, I think the idea is kind of you have a graph where maybe with literature references, and there could be a way that I can just kind of pull this into my graph. You do your data loading, however that works, and just connect to your graph and pull data.
Yeah. So in the beginning, we used that for debugging purposes because our database was way too big to debug on that and to reinitialize it when we broke the database. And we came up with an idea of just exporting a subgraph can be only a part of the genes, a part of the proteins, a part of the whatever, and then import that into your, let's say, on your local machine. But I could also see the same, let's say, if we have the same understanding of a gene and a transcript and a protein, like in textbooks or in any other scientific paper, I guess this graph model could be reused in other areas, and not only in healthcare, but also in biotech, right? So we had a very nice presentation by Clariant, who have a completely different use case, but we are talking about the same things. And I think you mentioned a really good point, post-translational modification. would be one of the more complex things to represent a protein, right? This is what we currently do not tackle so far, but this is a very important point if you talk about active proteins or inactive proteins or ubiquitized proteins. That is a big difference of a protein, right? But if you have some maybe an expertise where we have relationships and data integration processes on the entity protein, that could be an on top, let's say, or another level in the graph model. And that would be an interesting idea to discuss that and maybe have several layers in one graph database. And depending on your query or your application, we are traversing the one or the other subgraph or only specific relationships. So that could also be a possibility.
I agree that it is a complex topic in the end when you are considering post-translation modification on proteins and the epigenetics. And there are things which you can add on to graph. And like I said, so of course, a basic structure of the graph is always there. But adding on to it, actually, this is something which we are currently working on carrying. Because we would like to actually bring this sequence information actually in the graph. And of course, so we have the granularity until one gene results into multiple transcripts and could result into multiple proteins, and variants can actually attach to different transcripts then. So we have that kind of granularity already, but that is indeed a good thing about modification, because we are going to incorporate, or we are incorporating proteomics data there that could be identified, whether a protein is methylated, acetylated, or any kind of information where it is. I think a very good idea we have discussed is always creating a separate node which connects to that protein and give information there. So you have the information accessible, but it will not hinder if you want to query something which is not that fine-grained.
So we identified a couple of ways, and so first of all, areas in our graph where sharing makes sense, and also ways how we can share. So Alex and Rita, for you, the sharing should be simple, right? Because you work for public research institutions. Jeremy and Peyush, you don't. You work for a pharma company and a chemical company. So could you maybe share some insights or your general ideas? How easy is it for you to share details of your graph, and how much does the business needs, how much does it hold you back in terms of sharing?
Basically, I have to ask internally if it's possible, but for me, there are no problems, because it's by sharing that we are growing, basically. And maybe so some. It's always it was the same question for all the talks about refreshing data, about data injection, and so on. And we are currently using a Python framework which is named Kedro, which is really interesting to separate into different kind of pipeline specific way of ingesting data. And for the graph data model, it's always a good. It's not an easy question. Does it better to create and to try to create a unified knowledge graph to represent all the biology and the connected domains such as chemicals and so on? Or is it better to create a smaller graph, but that can ask specific questions? Here at Servier, we are in this way. So we have specific therapeutic questions. We are building a data model. And if the new data model can be incorporated easily into the big Pegasus, it's okay. But we can see a separate graph to answer separate questions as well.
I can give my 2 cents also here about sharing in the field. So of course, coming from industry, it is a bit harder sometimes to share all the information. But of course, this is something which we are looking forward to and looking into it. I was having this talk with Frank, our head of group CB, computational biology, and this sounds like an idea, because so at least I know from another projects, so they have been in a consortium where they give their ideas and they share ideas. So I think something like this could be possible. Of course, unless it's a sensitive information for company, everything should be possible. Means, of course, it means we came here today, we presented how we are working with our graphs, and actually the idea how the graph works like. So I think this is something which should be fine. But of course, this has to be seen. Maybe we can create a consortium in that way.
Yeah, definitely. I still think or hope that the health eco community might become a place where we can exchange those ideas, can share our subgraphs, and discuss methods how to do that. So I would like to thank all 4 of you for coming back and kind of taking the next step and discussing specific ideas, how we can share, what we can share, and how we can approach that. And I think we had a very, very good workshop here. 13 talks in total, lots of interesting topics, and obviously a lot of overlap. So like we mentioned in the beginning, we definitely want to turn this into a yearly event and continue. And we will also follow up with a survey for our presenters and attendees, of course, because we want to collect some input. How did you like the workshop? Are there any unmet needs, things we have to discuss? And I think for us, the next steps will be to figure out which areas are interesting for you. GDS, infrastructure question, data sharing, and so on, and see if we can provide more workshops and insights and so on. So thanks again to all 4 of you. Thanks to all the speakers if you're still listening. I think we had a great workshop. Alex, do you want to come in again? We had a fantastic workshop, so many interesting talks, so many interesting applications. I don't know what to say.
Yeah, no, I think thank you all again from Neo4j to all of you speaking, sharing your experiences and sharing your work with the wider community. I think these 2 half days were really, really insightful. Lots of great presentations. Today, I think that the topic was more on the knowledge graph side of things. Yesterday, it was with the sharing the visualization or the connected data approaches from lifelike, for example, as a session yesterday, still kept in mind, and today Alexander presenting the DZD knowledge graph, as well as others, Davide, for example, how to boost up a knowledge graph and how to get more information out of it. So I think that was really, really good. I had a good time. I enjoyed it a lot. We saw a lot of questions, so that's also always a good sign. People interacting. So I hope you all enjoyed this time. And like you said, Martin, I think we want to try to keep doing this, to do another session next year, maybe even on site somewhere possible. That would be cool to even do more sharing and more exchanging. Maybe have working groups even, or something like this. I don't know, but there could be ways to make this happen. So I definitely would like to see it again.
Absolutely. So I have nothing more to add. It was great.
All right. Well then, everybody, thank you again for joining, for watching, for presenting. And we'll see you again at another session soon.
And all the recordings will be available in a couple of days, right?
Exactly. Yes. Yes. Yeah.
Fantastic. Thank you so much.
Bye.
Have a nice day.